Okay. So first, good afternoon, everyone. Thank you for your time and working and collaborating with us to build a community of practice around team science here at NIH. My name is Corey Kelly, and I'm a program officer here at NINDS. And today, along with my Combine program colleague, Karen David, we're honored to host the first NIH NINDS Team Science Seminar Series. And today, we're going to be focusing on team science theory and practice. We have an excellent agenda today. I'll start with just a brief introduction to the science of team science, followed by Karen discussing how we've applied the science of team science to build the Combine program here at NINDS. Then our first cohort of Combine awardees will present their transformative projects along with a brief Q&A. And then we have some guest speakers who will close out the seminar. Liz Allen will discuss credit assignment, followed by Alyssa Pacchini-Schaefer and Hannah Bayer, who each independently provide their own perspectives on developing and managing team science programs. Just some housekeeping items. The chat is not enabled, but the Q&A is enabled. And so if you have questions, just put them in the chat. We'll try to answer them live, over audio and video if possible. If not, we'll reply in the chat. And I'd just like to ask everyone, if you have like a very specific question, I'm doing this kind of research, I have this project, is it, a good fit for your program or something like that? I'd encourage you just to send an email to Karen and I directly, ninsteamscience@nih.gov. And after the call, I'll sit here and answer whatever emails that come in until I'm done for the day. So we'll make sure to get back to in prompt order. We also would like to ask everyone to save the date for our second NINDS Team Science Seminar, which will be December 12th from 12 to 3 p.m. The topic will be convergence and complexity science. For those familiar with the Combine program, the two things we kind of talk about, and Karen will talk about this more, is transformation and integration. And this will really get into the weeds on how you can, use systematic approaches to both, navigate through complex scientific problems as well as bring together your team in alignment to, achieve your goals. In addition, we'll also be expanding this seminar series to the rest of the NIH. We'll be inviting some program officers from other ICs to talk about the team science programs they have. We'll also be inviting some of you from the community to discuss your team science projects. And we already have two excellent speakers lined up. We have Alex Peterson from UC Merced. His research focuses on data-driven approaches to complexity science. And we have Sabrina Figueiredo from George Washington University to speak about program theory, which I have a couple slides on later today. So I'll get started and just kind of introduce the science of team science. I just want to say there's a lot on these slides. I'm not reading hardly any of it, but I wanted everyone to be able to come back and use these as a resource. I've included a lot of references. I do have to provide the caveat with except for a few of these references which were published in collaboration with the NIH. These are just references that I've personally found helpful in my own career and not otherwise endorsed by NIH or NINDS. And a lot of the speakers will be talking about these topics in more detail during their presentations today. We are recording this, as I mentioned. We will send a link to the recording to everyone who's registered, as well as upload the talk to the NINDS YouTube channel, and we'll also put the slides on our Combine website, and the event website, so you can go ahead and download those and have access to these links. So, what is team science? Team science refers to both the approach of conducting research in teams and the complex social, organizational, political, and technological milieux that heavily influence how that work occurs. And although my talk today will be primarily about team science, the Combine program specifically was created to harness the power of team science to achieve the most ambitious goals in neuroscience, to achieve transformative science, a paradigm shift, a revolution in our understanding of the brain. There's been a great many paradigm shifts in biomedical science throughout the course of history. Yet, despite standing on the shoulders of these giants, neuroscience in the brain still remains the great final frontier of biomedical science. And the Combine program was established for those investigators willing to take the risk of eschewing normal science, pushing the boundaries of our understanding, and achieving a paradigm shifting transformation. However, the most ambitious goals and challenges of modern neuroscience research are increasingly dependent on collaborative approaches that leverage not only diverse disciplines, perspectives, and expertise, but effective interactions. And while team science is still a burgeoning field, the roots of team science at NIH began in the 1990s here at the National Cancer Institute to seek transdisciplinary approaches to understand the myriad of factors that contribute to cancer pathology, treatment, and care. And we're honored to continue work alongside these NIH pioneers, Michelle Bennett, Amanda Vogel, Kara Hall, and continue to build a community of practice around team science here at the NIH and with our extramural community. As the field of team science was born and the best practices of team science began to be elucidated, what many consider to be the foundational text in team science and enhancing the effectiveness of team science was published by the National Academy in 2015. And Karen and I are happy to announce that we're working together with members of the community to publish a revised version of this in 2025 that will include more recent considerations such as virtual collaborations and diverse perspectives. Finally, in 2018, the International Network for the Society for the Science of Team Science was formed, INSITS, and continues to be the leading professional organization for team science. And as we build team science programs and projects, we can look towards this pioneering work for lessons learned. We found that facilitators of team science include beneficial attitudes and beliefs, effective team processes, brokering and bridge building activities, and specific funding initiative characteristics that include things like providing opportunities for developing a community of practice. While barriers include integration, discipline-based differences, project management, and the incentive and reward system. And we'll talk a lot about all of these today. So first and foremost, how can we build effective team science programs and projects? Well, we start with purposeful program theory. This is alignment between the problem statement, the research questions, the conceptual framework, the methodological approaches, and the outcomes. Simply, alignment allows for a stronger argument of your program or project's causality, that what you're doing is actually the driving factor for the change. And a powerful program theory tool and visualization method is the logic model, which demonstrates your theory of action and theory of change, moving from your inputs, your resources, your people, to your outputs, your activities, your participation, and finally your outcomes. And by using this type of model, you can integrate, you can identify assumptions and integrate key performance indicators to not only evaluate your progress as you're implementing your project, but also demonstrate the causality of your outcomes when you complete the project. And when we zoom out to the team level and we zoom out and look at teams through a systems lens, we've identified characteristics of successful multi-team systems that include goal hierarchy, inter-team interdependence, inter-team differentiation, boundary spanning communications, and inter-team leadership. A fundamental model in team science is Bruce Tuckman's stages of team science development. First, forming, when your team comes together, then storming, which is when you're figuring out how to do work together, norming, which is when you're actually starting to do the work, and then performing, which is when you're actually kind of in the flow and the team's working really well together. And then, of course, adjourning, when the team finally ends. And it's important to note that teams should come together intentfully and then adjourn when they've achieved their goal. A critical component of team science is the inclusion of diverse perspectives. A fundamental tenet of team science is the concept that despite our best intentions, everyone will always have bias. And but by bringing together diverse perspectives, the impact of these residual biases is mitigated, and our findings will be more generalizable. Bringing together diverse perspectives requires not only understanding which perspectives are required to achieve the team goal, but also how to manage conflict and create common ground. As you look to form your team, define clear organizational structure, team composition, and roles. We say that the organization and the structure enables the people and the roles to utilize the platform and the processes to produce meaningful products. In the same light as shared vision, contributions, and decision-making. Last, conflict is normal and often inevitable. Having conflict resolution plans in place prospectively is far more effective than trying to deal with conflict in the heat of the moment. An excellent way to support these practices is to generate a collaboration agreement upon team formation. Data management and sharing is already a complex and sometimes laborious task that is further complicated in large collaborations. First and foremost, consider how you may harmonize outcome data from the various disciplines and methods contributing to your project. Identify cross-institutional barriers and have the requisite agreements in place prior to starting the work. And last, in my opinion, perhaps most importantly, plan prospectively. The worst thing that can happen is you reach the end of your grant and realize you can't harmonize your data in a meaningful way. Once the team is established and work begins, agile resource forecasting allocation is critical in the dynamic environment of team science. As I mentioned earlier, the logic model, combined with frequent communication, provides measures to inform the necessitated agile resource allocation that is necessary to achieve complex science. Moreover, the use of team science tools such as Gantt charts and Kanban boards can enhance team science or team effectiveness. Coordination, communication, knowledge are cornerstones of any scientific project, but particularly for team science. First, avoid cross-disciplinary crosstalk when people from different fields are saying the same words but meaning different things. Communicate and clarify. A dialogical environment, a culture of interactions that is founded on conversations between people rather than the more traditional dictation between a team leader and a team member, is critical for team science. Brokering activities that are specifically meant to facilitate conversations and transfer of knowledge, as opposed to activities simply for the dissemination of project information and steps, are an effective approach. And we encourage you to include trainees in these conversations, not only these conversations, but leadership conversations to better enable them as future team scientist leaders. And as teams begin to generate scientific products, credit assignment is often a source of acute conflict in scientific teams. Especially in the era of big teams, big data, multi-omics, it is essential to utilize and adapt a credit assignment taxonomy that prospectively defines the roles and effort that warrants credit on scientific products. We have the privilege today of having Liz Allen as a guest speaker, who developed the credit assignment taxonomy, which we cite frequently. Another pressing topic in team science right now is recognition of team science and performance and promotion. RO1s and first author papers aren't necessarily the primary measure for success in team science. So we need to begin to weigh the merit of team science alongside these traditional measures. It's really a call to action for scientists, administrators, and including us here at the NIH, whether you're leading a lab, a department, or an institute, to recognize and celebrate the successes of collaboration just as much as individual performance. In summary, how do we develop and conduct team science to achieve meaningful change? We start with purposeful program theory to establish probable causality. We then rely on implementation and dissemination to ensure fidelity of the program and uptake and translation of the findings. And we use team science to glue these together, ensuring both that we have the right team to achieve the goal and that this team can work together effectively. And now I'll hand it off to my program co-lead, Karen, to talk about how we've applied these lessons and key tenets to developing the team science and the combined program at NINDS.
Sounds good. Thank you, Corey. Is my slide showing okay? Great. Thank you. Thank you, everyone, for joining us. I'm Karen David. I'm a program director for the Brain Initiative. And I'm here to talk about our thought process in creating our new team science program at NINDS to promote transdisciplinary collaborations. So Corey, Kelly, and I co-lead the RM1 combined program. And in partnership with our NINDS program directors, we are happy to serve as a resource to potential applicants about this program. So for today, I'm going to be describing a general way of thinking about how NIH supports team science, and then pivot to describing how NINDS designed the new RM1 combined program by taking into account the opportunities and challenges that come with team science, which you've heard a little bit from Corey's presentation. But first, why collaborate? We're inspired by the team science culture from the Brain Initiative, as noted in this quote from the Brain 2025 report. And as we've seen from the acknowledgement sections of presentations, as well as published papers with the long list of authors, science is hardly a single person endeavor. Especially these days where science is advancing fast with new knowledge, data sets, and new approaches, it will likely take a team science approach to pursue challenging problems in neuroscience research in a way that's comprehensive and rigorous. So we can imagine what new and challenging questions that we can now ask if we combine forces, for example, between the tool builders with the tool users, experimentalists with computational neuroscientists, those studying the periphery with those looking at central circuits, looking at across levels from molecular to systems neuroscience, and also across species. And so at the NIH, what kind of opportunities are out there that will support collaborative efforts? I would note that team science funding is just one dimension of funding at the NIH, and our goal is to value and balance different scales of efforts from individual labs to different size teams. Typically for team science, there are two general ways by which the NIH supports collaborations. One is through the parent R01. And these R01s can be from a single lab, or it could be a multi-PI effort, meaning it's a collaborative effort across more than one PI with a significant role in the project. Now, some institutes have programs that support a larger scale of team science. And here you'll have multiple projects, usually from 3 to 5, and supported by multiple cores. And this type of structure usually comes in different names or mechanisms as listed here. Okay, so with these current ways of supporting teams, NINDS went on to ask, how can we further optimize how we support team science? So the general approach that we took at NINDS to address this question was to survey different ways to support team science within the NIH and also outside the NIH. We took note of the features that we liked, choosing components that will overall increase the likelihood of teams in succeeding by preparing them for the challenges ahead. For example, we found that frequently through these mechanisms that the teams tend to just operate in parallel without really the need to integrate or combine efforts. Some projects would have been fine to have been funded just as multiple separate R01s. And as such, for the RM1 Combined program, we require a single focused goal that can only be achieved by combining the efforts and diverse expertise of the team members. Also, team science is a social activity, and poor team management will sink the ship. As such, for the RM1 Combined program, we, meaning program and reviewers, pay attention to the components of the team management plan, and I'll be describing what those components are in the subsequent slides. We also encourage the inclusion of resources and personnel for managing and coordinating the team. Lastly, the success of the team effort hinges on the diverse perspectives that are included. And towards this, we borrowed from the BRAIN initiative this requirement of a plan for enhancing diverse perspectives, which I'll also be talking shortly. And as a result, NINDS adds a new structure of supporting team science here on the right. And to differentiate that from the other team structures that I mentioned before, to summarize, we have the program project on the left that is multi-component. And here, integration of effort is challenging because we usually end up with parallel efforts in the different components. In the parent R01, full integration remains a challenge because review or study sections tend to prefer independent aims. But in the RM1 Combined program, where our goal is full integration, we want pieces and efforts that are fully integrated and interdependent and that are essential to the single focus goal or aim. In general, we envision the ideal RM1 Combined program to have a single focused transformative goal that can only be achieved by the combined efforts by 3 to 6 PIs with distinct expertise, and that this goal is achievable within the funding duration. We encourage efforts that push boundaries in a way that balances risk, reward, and feasibility. And we require that the science is supported by a thoughtful plan for managing the team and incorporating diverse perspectives. So I'd like to call your attention to the 3 documents or attachments that we require in the RM1 Combined program so that the PIs can anticipate and prepare for the team science challenges that I've noted before. So one attachment that we ask for is the team management plan. And to craft this plan, we encourage applicants to consult this team science field guide developed by NCI. So for example, Corey mentioned about the collaboration agreement template, and we have that template noted in this field guide. Another consideration for teams is the inclusion of resources or personnel who will be needed to integrate efforts across the team members. And here, the PIs can include a justification of a program manager. And if integration of data is needed across team members, teams can also request for staff to help with this. So one of the team management plan considerations that I also would like to highlight is the credit assignment component, which you've heard a little bit from Corey. And this is tricky in team science settings because the incentive structure in science remains individual focused, with mainly the first and last authors given the primary credit. I'd like to note several articles that mention this issue. One is from our experience of funding team science of different scales and sizes through the BRAIN initiative. And we have Hannah Bayer from one of our brain-funded teams talking to us today about managing large-scale teams. Now, the transmitter, which is a publication supported by the Simons Foundation, and we're grateful to have Elisa Schaefer from Simons Foundation to speak with us today. So this recent article from the transmitter notes about the need for better ways of noting team contributor roles. So towards this, we have Liz Allen talking about the tools such as the credit taxonomy. And lastly, this article at the bottom notes about how academia can do better in recognizing team science contributions. So another attachment that we asked for in the RM1 Combined program is that they describe the timeline as well as the benchmarks for success. So reviewers use this information to assess feasibility as well as the degree of integration and necessity of each team member's contribution. And the last attachment that we asked for is the plan for enhancing diverse perspectives, which is pioneered by the BRAIN Initiative. So this plan is a summary of strategies to advance the merit of the science through enhanced inclusivity. So one thing I would note here is that our definition of diverse perspectives is broad and could be a variety of this. It could be who is doing the research. So the diverse perspectives could be coming from the difference in expertise, career stages, and so on. Or it could be who's participating in the research, or it could be in the diverse types of organizations and institutions. Okay, to close, if you're interested in this program, I recommend reaching out to your program officers, especially to check whether your topic of interest is within NINDS mission. And in our RM1 Combine website, we've also included a summary of these requirements, as well as the link to our applicant webinar, where we went through these requirements in more detail. So we're so excited to welcome our first cohort of our RM1 Combine Awards. And these projects cover topics that span basic, translational, and disease. And now you'll be hearing a little bit more about these projects in our next session, which Corey will then introduce. Thank you.
Yes, it's my honor to introduce our first cohort of Combine awardees. We're excited to hear about the transformative work you're doing. These grantees are just in the first year of their work, so very early on in these projects. And we are going to start with Dr. Juan Pascal, who will be discussing their project, Brain Glucose Deficiency Mechanisms and Modulation.
Well, thank you very much, Corey and Karen, for the great introduction. I have to say it's been a joy working with you and look forward to much more of that. Can you see and hear me okay? Is everything fine?
Yep, you're good.
Okay, great. So this is the title of the project. We deal with the brain glucose transfer defect, GLUT1. I have to apologize in advance because we have 80 scientists, 400 people, and about 120 patients right here, right now in Texas. We're collecting samples from the NM video and whatnot, so we can do some more work. So I might get called away later today to consent some of the participant subjects. There are three goals in the project. There's really one specific aim, but three postulates that cement what we do. First of all, we think there is inhibitory cell failure as opposed to excitatory cell failure in metabolic disease, especially in this metabolic disease, which alters specific circuit activity. Number two, we think there ought to be a way to observe this inhibitory cell dysfunction in individuals. And number three, we think they should be modulated through a variety of means for therapeutic benefit. So the basic underlying assumption is that this is a excitatory cell in green, there is an inhibitory cell in red. Excitation comes from below in the way of afferents, and this inhibitory cell will receive part of the excitation and therefore consequently inhibiting the excitatory cell, which is a triangle. And we think as a broad principle that in a number of these diseases that have to do with metabolism failure, it's actually that particular one synapse that's a primary fault. And that disinhibition or lack of putting the brakes on the circuit arises as a result of a complicated process which involves from the blood where glucose is really coming in from to the glial cell to the neuron, which is the excitable component, of course. And this whole system is quite complex because we don't fully know the elements and how they fully interplay with one another. So therefore, the approach has been to take a reductionist view. And the way we do that is to divide it into transfer, into metabolism, and to excitation. On the left, we consider the transfer phenomenon, so blood substrates and also red blood cell substrates, getting into the brain in blue, getting into the Krebs cycle in yellow, and eventually giving rise to GABA and glutamate, which are the key molecules. And that has to translate into excitability. And of course, there has to be a balance. On the far right, I have inhibition in red. I have excitation in brown. What we call GFO happens to be gamma frequency activity, which can be determined in a person through electroencephalogram. What I call LFO happens to be low frequency brain oscillatory activity. And so we take that ratio as an index of network state. If you look at the left, in purple, GOI, it's a measure that we call glucose oxidative index, which is the amount of glucose that's available to the brain that will be oxidized in the Krebs cycle. So we felt that if we develop a method or have a handle on these two values, which are quantitative, we ought to be able to, number one, define this disease and other conditions with inhibitory cell failure. Number two, we should be able to monitor treatment when appropriate. For that purpose, we have a team. Obviously, this cannot be done by a single laboratory. We have wonderful collaborators. They're based at UT Dallas. They're based at University of Florida and at UT Southwestern. Everybody has a different degree of expertise. Everybody deals with a different biological complexity, chemical complexity, engineering complexity. And so the way we have done it is to harmonize a number of approaches. In green, you have primarily rodent studies. In red, you have primarily human studies. And in purple, you have things that are shared by both. And the challenge and the point of the team project is to make all of the things harmonized or converged into a continuum, so we can go from molecular events all the way to the behavior of the person who's sick, who may be treated, and have an outcome that's meaningful to the individual. That's the summary. In terms of productivity, things that we have done, this is an NMR spectrum of a human brain after an infusion of C13 labeled glucose. And after some time, there will be brain metabolism and flux, and that will give rise to a variety of carbons that happen to be enriched in that C13 arising from glucose. What I'm showing in here in blue happens to be a volunteer who's actually a lab member. And then in brown, you have a patient with a GLUT1 deficiency, and then another one with GLUT1 deficiency. And what's really important in this whole spectrum is what's highlighted in green, which is bicarbonate. That was a surprise. It happens to be the very first metabolite that appears in the human brain as a result of a glucose entry into the brain. Why bicarbonate? Because the Krebs cycle will generate CO2. The CO2 will be eventually exhaled as we breathe, but that CO2 will combine with water through carbonic anhydrase and make bicarbonate. So it's a fairly simple readout of Krebs cycle velocity. And now we know that in a normal subject in blue, if you call that, we call that resonance 100%, or call it normal, if you will. Look at the two patients. One of them had 49%, one of them had 50%. So Krebs cycle velocity in terms of flux likely will be 50% reduced because of these hyperinsufficiency mutations. So we're quite comfortable we can proceed and develop this method. In terms of the excitability, we began from the brain slice, and this is a very complicated slide, and I apologize. So you're looking at the distribution of oscillations within a thalamocortical brain slice. So a rodent slice, it's mutated or not, wild type, and you have the thalamus and the cortex are connected, and they're perfused with high glucose, and then they're perfused with low glucose. Important point here, it was highlighted in red, which happens to be the high frequency oscillations. When we apply glucose in brown, the high frequency oscillations increase, as shown here in panel B. When that happens to the wild type, applied to the wild type, it's really no significant change. So we can manipulate, again, the high frequency oscillations that arise, perhaps from inhibitory cell activity, divide that by the low frequency activity, which is more related to excitation. And so therefore, we have a measurement of this low to gamma oscillatory activity in the brain slice. When we apply that to the human brain, this happens to be a composite electroencephalogram of 27 subjects with glucose transport deficiency. And you're looking at a 4-hour time frame in the electroencephalogram. They wake up, they have in blue delta waves, which are slow activity. On the right panel, you have gamma activity, which again is inhibitory cell related to some degree. And when you apply a metabolic fuel given by mouth, we see changes at about 6 a.m. in the bottom. We see changes in the delta activity, which happens to decrease, and we see changes in the gamma high frequency activity, which do happen to increase as hypothesized by palliating or mitigating the inhibitory cell failure. Again, these are individuals who have the defect, who have been treated as a proof of principle. So we look forward to continue, and we'll be glad to take any comments.
Thank you, Juan. If anyone has questions, you can place them in the chat. Okay, if not, we'll just take a brief break while we get the next presentation set up. Dr. Bessie, are you going to be presenting your work, or is Rafa?
Rafa's going to do the intro, and I think the plan is all of us are going to present.
Okay, wonderful.
I think they're joining in. I see a few more.
Okay, no worries. Otherwise, we can just switch them up as well.
Okay.
Sorry, there's
see
a question for Juan Pascual.
Okay, yeah. Juan, can you see that question?
Yeah, I can see that question. The question is, how do these high frequency oscillations correlate with PET? Well, I mean, that's a fundamental important question, because in neurology, neurobiology, the main issue is the harmonization of time scales, time domains. It's a pending subject in neurology, neuroscience. You gotta consider the frequency of what you're looking at. These high frequency oscillations happen at about 40, 50 times per second. A PET scan, it's about a 30-minute composite image of glucose accumulation within the brain in an anatomical fashion. It's not even uptake, it's not even metabolism, it's glucose accumulation, influx minus efflux, signal intensity. So the correlation is such that if you think of inhibitory activity causing thalamocortical oscillations, the depressed inhibitory activity is causing thalamocortical oscillations, which is common type of absence epilepsy, which this disease is an absence epilepsy. We do find by fluorodeoxy PET scanning in people depressed thalamocortical signal with fluorodeoxy glucose. But again, entirely different time scales, so no clear correlation, just an association. But yes, it goes with the concept that you have this cell dysfunction in the thalamocortical circuit. Thank you.
Thanks, Juan. And there's another question. Could you please discuss challenges in integrating across the institutions that you may have faced?
Yeah, we're geographically located in different parts of the country, because I moved to New York about a month ago. So add to the complexity of, and the whole lab moved, and we move mice, we move equipment, we move 6 people. We have to comply with another set of regulations. So, if anything, we had a complicated deal. The way to mitigate that issue have been to, number one, I retain appointments at my former institution, and I gain new appointments at the other institutions. So for me, I'm part of the thesis committee at all of the other institutions. So it's seamless for me to actually fly in and be physically there. We found there's a huge difference. So we meet every other week by video, all of us, but in reality, there hasn't been an easy way for us to replace physical presence. So going there, being part of that helps. The second thing we do is we get the trainees to do things together virtually. The article that you saw, the first author is a PhD student making a lot of progress. The senior author happens to be a person who's soon to be a faculty in a few months, so making a lot of progress. And they all got together and collaborated with an online data share tool. So it's been facilitated by technology, but it's also being facilitated by physical presence. It hasn't been too much of a problem. Part of the reason too, I should be clear, is that a lot of the team predated the RM1. So we already have collaborations, and we already knew how everybody thought, and we already knew the key people. So that made it easier. If it's a brand new team, my only advice to you would be to try to emphasize more physical presence, maybe a symposium, something like that, which we're doing as well. That's what we're doing right here today. The whole team is here, and we brought in another 75 plus scientists from other fields. So, hopefully maybe they will join the next team.
Thanks, Juan. Could you maybe perhaps describe how the individual contributors came together to kind of define what your single goal was, or vice versa?
It was not trivial, because every one of us have the capacity, like all of you on the call who are scientists, every one of us has a capacity to produce immense amounts of data. We are all pretty seasoned scientists in our own way, and there's no question we can produce. The question is, produce something that's really relevant to the one aim, to the goal. Prune out a lot of the redundancy, a lot of the things are not very meaningful, and then put that in the back burner and focus on the one thing. So it was extremely helpful for us to have the milestones agreed upon. One of the components is the milestones, and it forces you to really think about what's going to happen here and then, and we justify why we agreed on that one milestone and the next one and the next one. So it's not just for the sake of progress reporting, which is important, but also for the sake of why did we choose providing the moving rodent data with single cell activity to the team that does EEG by month number 9. There was a debate on why that would be the right thing to do and what would be the time frame realistically to get. It forced everybody to comprehend the complexities that everybody else had to accommodate just so they can provide that data in that time frame. So I think the milestones were crucial for us to be able to get people to come together. The second thing is we rewrote our authorship criteria. We harmonize, very diverse. Everybody will have an authorship criteria document, and we all use different things. We actually have a couple of meetings, the whole team, to agree on who's going to be an author, who deserves to be acknowledged, who will be the first one, the last one, in an equitable fashion. And if you have that up front, in our case, it's been very, very smooth, because there's no question who's going to own which portion of the work. And finally, I should say to all of us, and I'll say this, maybe inappropriate to say this in front of everybody, but the role of PIs is to really be in the background, as opposed to be so much in the front. So all of us are taking an extremely supportive approach, but we don't want to be known or recognized as the lead for anything. Our presentations here yesterday and today are mainly done by students, in fact. We went through them, we helped them, we coached them, whatever it needs, but we don't want to be identified as the anything. I'm done with that. I have too much of that. I want to be in the background. So a combination of being humble, combination of supporting people, but having the milestones and understanding what it takes for somebody to provide you with their milestone made a huge difference.
All right. Thank you, Juan. There's a couple more questions in the chat if maybe you want to get to them while we move on to the next presentation. Next up, we have Dr. Yusta, Basu, and Segev, who will be talking about their combined project, re-examining the role of dendrites in neuronal function.
Hi, everyone. Can you hear me well? I may have bandwidth problems, so if I happen to freeze, Yayita, please, could you take over if there's a problem with the connection?
Do you want me to share the presentation? Or?
I think let me try that, but I will turn off my my camera to increase my bandwidth. So I hope you can see our first slide. Just let me know if you can. No. So so we're after something absolutely fundamental in neuroscience, which is the question of what is the functional unit of the neuron. And I think it's fair to say that for the last 100 years, in the time of Cajal, people have assumed that the fundamental unit of the neuron is the synapse. And if you look at neuroscience today, the majority of projects, it's based on the study of synapses, whether they go up and down, and how they can relate to learning, to pathology, to all kinds of things. At the same time, these synapses are on these dendritic spines that are positioned in these dendrites. As you know, neurons are like trees, no? And the dendrites are branches. And everyone pretty much assumed that the dendrites were the place where you put your synapses. It's like parking space for synapses. But already maybe 30 years ago, people starting to notice that the dendrites were active, and that you could have dendrites that would fire. One branch of the tree would fire, like is illustrated in this drawing, like this one in orange, but not the rest of the dendritic tree. No? And this has been traditionally put under the rug in neuroscience. We all knew that there are data that showed that dendritic branches have activity, but people said, well, maybe they have activity, but really the synapses is the ones who are doing the job. Anyway, now with the last years, with the introduction of genetically encoded voltage indicators, imaging in vivo of these dendrites in the mouse cortex has demonstrated that these dendritic spikes are much more common than we thought. And in fact, they could be the conductors of the neuronal symphony, to the point that we suspect they could be the functional units of the neuron. So that's the question. And if we're right, we turn neuroscience upside down. If we're wrong, at least we gave it a good shot. And to give it a good shot, what we're trying to do with our team is study the same exact neuron, the same neuronal cell type in the mouse visual cortex, a genetically defined neuron, and study their dendrites, but do it in a complete fashion, actually following Sidney Brenner's motto of complete, accurate, and permanent science. And we're 6 teams of people. Michael Lin builds genetically encoded voltage indicators to map the activity of these dendrites in vivo, of this particular cell type in the mouse cortex. Our team uses Michael's genetic indicators to map and measure the activity of these spines and dendrites in vivo. Yayita Pasu's team uses electrode recordings from these dendrites to characterize these dendritic spikes and understand the biophysics and the mechanism, how they're generated. Eli Medivi's team uses modern expansion microscopy and proteomics to map the receptors present in these dendrites, and Jeff Lichtman's lab, ultrastructural connectomics, to reconstruct the morphology of these dendrites. So all these data sets are then sent to Idan Segev's lab in Israel, and Idan is actually the world's expert at making computational models of dendrites. So, so what we want to do is essentially, we call this the Rosetta Stone project, to essentially provide a data set for the world, for the field, in which for one particular neuronal cell type, we know everything we can know in a complete, accurate, and permanent fashion about these dendrites. And with this data set, we want to test a hypothesis that, hey, what is the functional unit of the neuron? Is it the dendrite or is it the synapse? And very quickly, I have slides. Michael, if you're in the call, would you like to very briefly talk about this slide? Maybe he's, yeah.
Got it. Yeah, thank you for unmuting me. Yes, hi. So our role in the project is to develop the voltage indicators that will allow visualizing dendritic voltages in vivo. And along these lines, we have been developing a family of fluorescent voltage indicators named the ASAPs. And this is just an example of our latest ASAP, where In the top, you see an electrophysiology trace, and then below you see examples of electrical impulses visualized by fluorescence. And you could see that basically we could track the origin of dendritic electrical impulses from their points of origin. For example, in there's one impulse that starts from the red region of interest number 4 and propagates down the dendrite to the cell body down to region number 1, the gray region. And so we can see an excitatory postsynaptic potential resulting from probably the reception of a single synaptic vesicle event causing a small electrical impulse that then travels down the dendrite to the cell body. This is an example in vitro, but we're
Michael.
Yes. But
No,
the other members of the
quick
group
because
will be using this tool to study dendritic propagation in vivo. That's it for me.
Yeah, this is an example of using Michael's tool, the one he just described, to map in vivo the activity of all the dendrites in this particular neuron. You can see here on the right examples of the voltage imaging from this dendrite. Yayita, you want to talk about your just very briefly your group will do or is doing?
So one of the things our lab can do is go and record from these submicron thin dendrites. And here you're seeing on the left side of the slide here in blue, one of the first recordings from layer 2-3 visual cortex pyramidal neurons in the slice. And you can see here, we can record the intrinsic firing properties of the neurons, as well as stimulate different pathways like layer 1 and layer 2 or layer 5, and see what the synaptic responses look like. And we can compare that to what somatic recordings look like. So that's very powerful because then we can directly get intracellular conductances as well as synaptic properties mapped onto the dendrite. Now, another thing we're doing very closely with Michael is establishing the ground truth, so we could patch from the soma and the dendrites and see how linear is the voltage imaging signal as compared to the voltage electrophysiology signal. And you can see one of our first ASAP5 traces here in green. And finally, an initiative that we had started as part of our Circuits Brain Initiative project. And now this project is allowing us to apply this toolbox. It's an automated dendritic ROI segmentation toolbox because this did not exist. People had to manually segment ROIs of dendrites, and dendrites are very elaborate, so it's a tedious process. So we used data from Rafa's lab, and we could ROI segment them automatically using this toolbox, which we want to make available to the community. I'd have to say one last thing. Rafa and our labs are next door to each other practically in Manhattan. So we intend to bring this, our combined collaboration, on the grounds by using animals that were recorded in Rafa's lab in vivo and then recording from them in slices. So that's one thing we can do to integrate better.
Ellie, if you're in the call, could you very briefly describe this slide?
Giving you co-host.
All right. So
an additional piece of information that we need in terms of how dendrites are computing is not just the activity across these dendrites, but the location of the synapses and the flavor of the synapses, whether they're excitatory and inhibitory, and who their partners are. And so we've developed a combination of genetic and molecular tools seen on the left here, where we can map all the excitatory or inhibitory neurons, synapses onto a neuron and who their afferent input is. Then we can image these neurons in vivo and look at the dynamics of the synapses. And then post hoc, we do an expansion and clearing where we can validate the synapses, and we could also then get information on synaptic size and synaptic content in terms of receptors. And this labeling method will also allow us to send labeled cells to Jeff Lichtman's group, which we'll talk about in the next slide, where they can have the additional information of the afferent inputs, which you normally don't get by EM.
Yeah, so Jeff couldn't join the call, but this is just to illustrate how they have an automatic pipeline that process data from this particular cell type in the cortex and identify with ultrastructure, reconstruct the entire dendritic tree and identify the axons that connect to every one of the synapses that Ellie was just talking about. And just to finish, Idan, would you like to talk about this final slide of our group?
Yes. So first of all, it's very exciting. So I'm very excited about this project and very happy to be part of it, really. So the first thing, of course, in order to integrate all the data that you heard from different labs, we have to build a pipeline that takes EM data, as you can see on the left, reconstruct a full neuron or represent a full neuron, which is next, and then really take this full neuron, the skeleton of this full neuron, including all the spines, and build a compartmental model in the right that will represent the whole cell. Then we can embed different information, physiological, imaging, and so forth. So we already actually finished this part almost. We are now testing. So, you know, I call it from EM to neuron simulation. So the tool is already ready almost. And we already started actually on the bottom to try to analyze what does it mean that these different spines, as you can see on the left, have different shapes, the synapses, the excitatory synapses on the head. And we are really focusing on the timing effect of the shape of the spine neck on the EPSP at the spine head. We found some interesting capabilities of the spine to track high frequencies if the spine neck is resistive enough. This is in the center. And so we are starting to really simulate realistic spines in realistic reconstructed neurons. As I said, very exciting.
Thank you. Sorry for going over time. Yeah.
Well, good. Thanks, Rafa and team. I'm just curious because you have all these multiple interesting components and pieces to your project. What do you put in place to make sure that you're all keeping track of each other's developments and making sure that you're heading towards the single goal?
Yeah, we have monthly meetings, which are quite, I would say, intense, I would say, and then constant communication between us. Jayita and I are on the same city, and we also share, this summer we're sharing a student. And Michael and I were already collaborating. In fact, we have a paper submitted together as we speak. And what really binds us together is that we're working with the exact same cell, the exact same cell type. And that's our goal, to deliver to you, to NINDS, and to the whole field of neuroscience, a Rosetta Stone database. And we call it Rosetta Stone, just like in the Rosetta Stone, you have the same message written in different languages. We're studying the same exact biological entity, which is the dendrites of this particular type of neuron. Study them from different angles: structural, molecular, physiological, biophysical, and computation. So that's really what binds us together, that we are the 6 crazy labs in the world that are focusing a lot of their effort in one particular cell type.
I'll just add two more things that Rafa has really driven forward. So we have, as a team, we have a Slack channel where all our trainees interact and exchange resources and information. And we also had one symposium already in December, and we plan to meet in October during SFN in person, the entire team. So not just the PIs, but all the trainees together. And we're going to present and discuss the data. And then in addition to these monthly meetings, each lab also has a roster to present data and do Q&A. So there's a lot of facilitation of cross-interaction. We've already exchanged a lot of reagents and resources. Ellie and Jeff are also in the same city. So there are some, lines of sample sharing and reagent sharing, but also resource and codes. And I think, Rafa, you wanted to talk about the data management.
Yeah, I think only if there's time and if there are questions. No, what?
If you really quick, it would be okay, and then we'll move on to Dr. Kroos's presentation.
Yeah, I think our plan is to deliver the data using not just a GUI interface, and we're working with the Allen Institute to do that, but also try out something that they're trying at the Allen, which is a knowledge graph, which is a way in which different data sets are interacting smartly with each other, a little bit like pulling on a thread. No? So this could be a perfect test case for the knowledge graph. Imagine that you're interested in a particular type of receptor, and you find through the internet, oh my God, it's in this dendrite number 5 that Ellie Nedivi just reconstructed. And you click on that link, and the knowledge graph takes you to the function of that dendrite, the ultrastructure of that dendrite, the biophysics of that dendrite, computational model, and suddenly you are sort of in a different world. So this is what we call the knowledge graph. So we hope to be able to do that. We're inviting the key person from the Allen Institute to our on-site visit, so to speak, during SFNA Chicago, in which the Korean Karen will be invited very soon. And and we also have, I just wanted to share the good news that we have also another symposium coming up on, I think it's December 12th on this year, a repeat of last year's symposium with 6 new speakers, and all agreed. So we're ready to go for this year as well.
All right, that's great news. I really enjoyed the conference last year. Look forward to it this year. Okay, thank you everyone on the team. Great presentation, great work you're doing. There is some questions in the chat. If the team wants to reply to those by typing in the answers, that would be great. Okay, we'll go ahead with the next presentation. We're going to take a break around 2:30 as well, just so everyone knows. So the next presentation, Dr. Kroos, Lopera, and Arboleda Velasquez, which will be discussing their project, Resilience to Cognitive Decline and Resistance to Alzheimer's Disease and Related Neurodegenerative Diseases in Individuals from Colombia with Autosomal Dominant Dementias.
Perfect. Can you see the screen?
Yep, you're good.
Perfect. Okay.
Thank you.
So, thank you again. My name is Jaquel Quiroz. I'm an associate professor at the Massachusetts General Hospital, and I'm presenting on behalf of the team that includes Dr. Francisco Lopera from the Universidad de Antioquia in Colombia, and Dr. José Arboleda Velázquez from the Skepenstein Research Institute. And you already heard the title. I know that it's a very long title. So we actually make it more friendly, and now we are called the PRADA project, that is the Protection Against Dementia in Antioquia, and this is how it's now in Colombia now. So just to give you some background, and you probably already know this, but this is kind of where we started from. We are interested in understanding what are the common protective factors across dementia. And for that, we are using, this is just an example of all the different etiologies that can explain dementia, being Alzheimer's the most common one, but also like Lewy body, vascular dementia, frontotemporal, and so far. And we also know that mixed dementia is something very common when we're talking about dementia. So in Colombia, I'm originally from Colombia, as well as my other 2 co-PIs. And in Colombia, we have very large extended families that are actually impacted with autosomal dominant mutations that lead to Alzheimer's disease, frontotemporal, Huntington, and of course, CADASIL as well. And this work with these families that is almost more than 30 years has led us to pretty much work with these families, identify who are the carriers and non-carriers, and also identify novel mutations that explain this disease, as well as some mutations that explain protection. So a few years ago, we started with the COLBUS, that is Colombia-Boston Stream Protection against Alzheimer's disease and dementia. And the main goal was to actually identify those that, even if they were destined to develop dementia, especially considering Alzheimer's disease due to a mutation, so they didn't at the expected age and they remain cognitively unimpaired until older ages. So with that, for instance, so far we have identified, and this is like putting together in the same region of Antioquia in Colombia, we have about 6,000 individuals that actually have belong to these families with a single mutation for early onset Alzheimer's disease. And out of those, about 1,000, oh my God, sorry. So 1,200 mutation carriers have the single mutation. And so far, we have identified a little more than 30 that are potentially protected. We also have families with NOTCH3 mutations, actually 3 different mutations in NOTCH that leads to CADASIL, that is the cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy. That is also, it leads to early onset vascular dementia. And so far we have about 200 individuals that actually carry these mutations, and out of 260 that are potentially, that were evaluated at 8 that are potentially protected. And for FTD, for temporal dementia, we also had large families that actually had mutations in several different genes, but for this one, we are focusing on the ones that have mutations in the MAPT. And so far, we have evaluated 30 individuals, and 2 that seems to be potentially protected. And those that are potentially protected are the focus of this investigation. So the first case that I actually reported at Primas led us to propose this grant. It was the case of Doña Lidia Piedrahita. You may have heard about her. It's the individual that we identified that belongs to these Colombian families with autosomal dominant Alzheimer's disease. And she was destined to develop Alzheimer's in her 40s, but she only started showing symptoms in her 70s, like 30 years, almost 30 years after the expected age of onset. In her brain, we observed that she had really high levels of amyloid pathology, but very limited levels of tau pathology and neurodegeneration. And we found that she was homozygous, as I said, for the APOE3 Christchurch mutation. And some of the pretty much the experimental work that has been done so far to understand these mechanisms of protection in her case, in particular in Christchurch, has shown that Christchurch is important for this binding between ApoE and a type of sugars that is called HSPGs. And also most recently done by other groups, Christchurch seems to impact the function and the response of microglia, especially as they suppress tau, serine, and beta amyloid. So we are still learning about the mechanism, but this is pretty much what we actually propose and wants to do across different dementias. So as an undergoing work, and this is something that it leads to the RM1, so we are interested in understanding the mechanisms of neuroprotection, particularly with Christchurch. We wanted to see if there was any gene dose protection, and of course, we're interested in understanding other SCAPs and other identifying other potential protective mutations. So just recently, actually last week, we published this report that it was, we went to the 2,000, about 2,000 individuals that we have in the autosomal dominant Alzheimer's disease, and we genotype and looking for Christchurch, and we found 121 members that actually had 1 copy of the variant, and 27 carriers that only have that have the 2 mutations, the Christchurch and the presenting one. And in this report, we actually did the analysis that led us to conclude that the Christchurch mutation carriers only with 1 copy have a 5-year delay in the onset of MCI, and also 4 years of delay for dementia compared to the non-carriers. And here I'm showing you in the right, sorry that it looks too small, that's pretty much the family tree or all those 121, but also in the bottom one, I'm showing you, you can see. And showing you here the neuroimaging of 1 individual that actually had really 2 mutations. And when he was 51 already, like a few years after the onset for this family, that is 44, he have similar levels of amyloid, a little bit more than the control, but he have a tau pathology when we actually very limited amounts of tau pathology compared to the control. So pretty much all of those lead us to the conclusion that Christchurch may in fact be protected in a gene dose matter. So for the RM1 premise, we are interested in defining pathways for cognitive resilience and resistance, as I mentioned before, that are across neurodegenerative diseases. So we are going to be going from normal aging, that are pretty much the controls, to the typical individuals that actually carry mutations for Alzheimer's disease, FTD, and CASI, and comparing those to those that are protected. And by protection, we define that as being either resistant or resilience to cognitive decline. So for the background design, as I mentioned earlier, so we have these individuals that we have already identified. We're gonna characterize those first. We propose to extend, as I mentioned, like we're gonna be looking at the other individuals that are protected that we haven't identified. We are interested in pretty much understanding the mechanisms of cognitive resilience, and with the main goal, and this is something that we can discuss later, to identify disease-specific mutations, genetic protective factors, and also some gene variants that can be common across dementias. So the significance of this, of course, I already mentioned, but it's also important to identify those individuals that may be protected. I personally believe that protection against dementia is going to be the future, and it's going to be the way to actually prevent a decline altogether. So we definitely need to understand their neuroprotection with more precision and trying to use that information to pass it to other individuals at increased risk. So we will test the hypothesis that I already said that, and we're going to identify and search for new variants in individuals that are protected for the other dementias as well. So once we identify that, and this is the part that we are integrating now, so pretty much once we identify those individuals, we will examine the relevance of those candidate variants using molecular and experimental studies. And in the future, once we have all of these, we plan to use IPS organized to better characterize the relevance of those gene variants. So sorry, I tried to do this the best that I could, but it's kind of like how we kind of like work together. So Dr. Lopera, that is the team in Colombia, they're starting with all the clinical and cognitive measures that are pretty much in these individuals that I said protected. So all the neuroimaging and biomarker characterization on these individuals, including neuroimaging blood and other measures as well, is something that my team is contributing. And Dr. Arboleda Velázquez, he's gonna be also doing, or he's already doing the genetic analysis and will be doing in the future the IPS work as well. So the way that we're trying to integrate this, I just anticipating some of the questions, like we actually meet bi-weekly, and we review the cases that we already think that are protected, and those samples are sent to Dr. Arboleda's lab for analysis. And once we have some candidates for a protective mutation, we go back to the family members and trying to better characterize those individuals. So for ongoing work, as I mentioned, we are not called product. What is in red is what we are doing so far. So we are in this stage that is team forming. So we're starting with the patients, identifying the potential patients, running all the clinical and biomarker examinations. And as I mentioned, like running the genomics in the individuals that we think that are protected. And also with a goal, or pretty much, and I like what Cory mentioned at the beginning, like this idea of creating community around this topic. We actually proposed the first Latin American symposium that is happening in August, when, of course, all of our teams are coming together, but we're also opening to the community, the scientific community, so they can learn what we are doing and they can get involved in the project as well. So we had the first one in the 22nd and 23rd of August. And with that, I would like to thank everybody in our teams and, of course, in the teams of my collaborators as well. Thank you.
You, Dr. Couros. Let's see, Q&A. There is a question in the chat for you. Will organoids be sufficient in uncovering these mechanisms? Is it possible to include animal models of this phenomenon?
Yes, yeah, IPSS is not going to be sufficient. It's just like a starting point and the same, like, because we propose it here, but what we have seen in the past, even with the Christchurch, like as I mentioned to better understand the underlying mechanisms, so probably animal models is going to be needed as well.
Thank you. All right. Thank you, Dr. Couros. I think it's your project is a great example of how you can bring diverse patient perspectives into the team as well. Okay, if you have, let's see, any other questions for Dr. Crows, please put them in the chat. And then we'll go ahead and move on to our next presentation. Doctors Cooper, Dixon, Lee, and Sardiello. And I believe we have a special guest as well. And this project is called a systems level approach to neuronopathic lysosomal storage disorders. All right. Go ahead when you're ready.
Hello, we are the team for Systems Level Approach to Neuronopathic Lysosomal Storage Disorders at Washington University School of Medicine in St. Louis, Missouri. Our project applies multi-omics techniques to study 2 inherited neurodegenerative lysosomal disorders of childhood, Sanfilippo-B syndrome and CLN2 neuronal ceroid lipofuscinosis.
Every cell in our body contains lysosomes, where proteins are broken down and recycled. This can go wrong, resulting in the buildup of storage material, but we don't think this kills cells. The lysosome is involved in many other processes essential for keeping cells alive, and this is especially important in the brain. We know that having faulty lysosomes leads to massive neuron loss, which also happens in many other CNS diseases. Our challenge is to properly understand lysosomal dysfunction and how this differs between disorders and cell types. Until recently, we weren't able to do this.
Our solution includes 3 separate but complementary steps. We will isolate lysosomes from affected tissues and cells to study what in their composition is critically different compared to healthy individuals. Based on these differences, we will identify and experimentally test specific biological pathways as candidate disease drivers. We will also analyze if enzyme deficiency affects the communication between lysosomes and the nucleus. We will focus on transcription factor EB by studying how lysosomal defects alter its post-translational modifications and transcriptional output. Then, we will restrict enzyme deficiency to neurons, astrocytes, or microglia to understand how each cell type dysfunction impacts on disease progression. We will identify the relevant players by analyzing pathological markers as well as lysosomal content and signaling.
Lysosome storage diseases and most human diseases arise from many factors, which is a combination of genetics, but also proteomics, metabolomics, and omics in general. Even we know that all of these factors are implicated in disease, so we don't know how they interact between them. Multiomics, which includes genetics, epigenomics, transcriptomics, proteomics, and metabolomics, can measure the intermediate molecular characteristics from a specific disease. We have been using multiomics approaches for multiple diseases, including lysosomal storage diseases, to not only identify those factors that are associated with disease, but also those that are part of the causal pathways, and they can be used as a potential biomarker and to identify potential targets.
After the omics data profiles are generated, we will integrate, analyze, and interpret the data using graph AI models to identify the key molecular targets and the causing pathways associated with the diseases.
Organelles like lysosomes can really tell the story of what's going inside these human cells that are sick. We have microscopes that can take beautiful images of these cells, but more importantly, we have analysis that can really capture the subtle signatures that make these cells the correlates of human disease. Therefore, we can use these cells to find treatments and to figure out when we fixed them and reverted the phenotypic signature back to normal again.
Most of the work in our lab focuses on developing treatments for Sanfilippo syndrome and similar lysosomal disorders where replacing the missing enzyme can be therapeutic. But for this project, we're using an enzyme for Sanfilippo-B syndrome that is tethered inside the lysosome of the cell to study how correcting disease in different cell types affects the disease process, so we can learn more about it.
Mass spectrometry offers precise identification and quantification of lysosomal proteins, detecting low-abundance proteins, modifications, and interactions. Combined with the bioinformatics, It provides a detailed view of the lysosomal proteome, essential for understanding lysosomal functions and diseases.
Part of our project is generating patient-derived cell lines and phenotype data to ensure that our findings relate directly to patients. We partner closely with patient advocacy groups to understand the unmet needs of patients. They help us contextualize the observations we make in the laboratory about the biology of these disorders into the reality they're facing every day.
The neuro-LSD community increasingly recognizes that these conditions are multisystemic, not solely neurological. WashU's research promises a comprehensive roadmap uncovering the foundations of these devastating diseases. This advanced research is pivotal for developing transformative therapies in CLN2 Batten and other LSDs, marking a shift in our approach to treatment.
We're so excited by this Team Science Award and what its findings could mean for families living with lysosomal diseases like Sanfilippo syndromes. There are still no effective approved treatments for any form of Sanfilippo syndrome. And so projects like these, which use a team science approach, have the potential to move the needle in ways that just haven't been possible in traditional research. Collaboration is really the key to building the innovative solutions and therapies our children urgently need.
Thanks for letting us tell you about our project. And please let us know if you have any questions.
Amazing, thank you. I should consult with you on all of my presentations. If you have any questions in the chat, Dr. Busher and I have joined us as well.
I think a lot of our team are on.
Yeah, if anybody needs co-hosts, just let me know. I tried to catch everyone.
How did you decide which particular lysosomal storage diseases to focus on? I don't know, John, you want to start with that one?
Yeah, well, what we want to do is essentially compare and contrast across two diseases which are thought to be similar, but actually have different underlying causes. They have different gene defects. The material that accumulates them is different. They have a different progression of their pathology. And we thought they would be good candidates for taking two diseases which on the surface should be similar, but actually will probably turn out to be quite different to one another.
Well, thank you very much for that very well-produced and informative presentation. I look forward to watching it again. Okay, if you have questions for Dr. Dixon and her team, please go ahead and put them in the chat. Otherwise, we'll go ahead and move on to the next presentation. Dr. Zayad, Lee, and I think Dr. Ad will be presenting today, and the title of this project is Targeting Cell Type Specific Disease Phenotypes to Promote CNS Repair.
Good afternoon, everybody. Do you see my screen? Yeah. Okay, so it's a pleasure to tell you today about our work where we're...
Maggie, you may want to, yep, that works.
Okay, yeah, thank you. Our focus really is spinal cord injury, and we are a group led by my lab at Georgetown and Dr. Jay Lee's lab at University of Miami and Dr. Keban Lee's lab at Rutgers. And what we're trying to do is to target specific cells after spinal cord injury. So you don't want to target all the different cells after spinal cord injury, just specific kind of cells that promote injury. Spinal cord injury, of course, is very common among males, and we all know that there are locomotor deficits. Importantly, there are no new medications for spinal cord injury. So that led us really to try to think outside of the box, look for new novel ideas and how to integrate our opportunities. So one opportunity that we had came from Jay Lee's lab, where in a mouse model of spinal cord injury, what they found is that macrophages become foamy and have these lipid droplets. And if you remove these foamy macrophages in vivo, then you can basically have functional recovery in animal models. So our main idea overall is to try to target these foamy macrophages using a strategy where we have nanoparticles that have small molecules inside these nanoparticles. And then the idea is that the nanoparticles that are developed by Kevan's lab have antibodies that specifically recognize the foamy macrophage. So how do we find these small molecules to target specifically these foamy macrophages? We, in our lab, working on glioblastoma, actually, we developed a novel platform called isosceles, which integrates small molecule information with disease signatures. So through the LINKS consortium, we had small molecules and information, how these small molecules induce transcriptional changes. So this is what we call a compound signature. And we integrate the compound signature with a disease signature from a specific cell, such as the foamy macrophage that I mentioned, or a fibroblast. So that's our first step. Our second step is to work with Kevan's lab to put these small molecules that we predicted into nanoparticles, and then to see specific nanoparticles that would target the foamy macrophage or fibroblast. And then eventually, in collaboration with Dr. Jay Lee's lab, actually test the nanoparticles in animal models of spinal cord injury. So this is really an integrated approach that required this mechanism because there's constant refinement of all the different steps. In terms of progress in the last year, we've been working closely with each other. One story that Dr. Lee had was about taurin-2, which affects the PI3 kinase pathway. And importantly, taurin-2 is a small molecule that reduces these foamy macrophages in spinal cord injury. So Jay's lab had a paper showing that this is beneficial in a spinal cord injury model. In my lab, we actually wanted to find out where the small molecule would target the different cells within a spinal cord. So we had Visium data looking at, this is an injured spinal cord, and looking at the different cells. We analyzed the different cells. And then we used our isosceles platform and integrated that with the spatial data and said, where would taurin-2 localize? And we found that taurin-2 would localize and target cells in this injury site as shown here. So that's one form of progress, and we're going through the different small molecules that we're predicting and then saying where in the injured spinal cord, where would they target and what kind of different cells would they target. Some other progress, Rishika, a bioinformatician in the lab, developed something, a shiny app called orthogonal, that we're making available, and we're sending out a publication soon about this. So the issue we have is that a lot of the LINKS 1000 data, all the LINKS 1000 data is in about human genes, but in the spinal cord injury model, we have rat models or we have mouse models. So how do you integrate this information? So without requiring coding, Rishika developed a shiny app where you can upload genes that you have for a disease signature from rat or mouse or zebrafish, and that would transform it to the human species to allow you to compare the different data sets very easily. As part of our DEI initiative and what we did, actually, we worked hard on this to develop a course for underrepresented minorities to learn the different coding that our group does as it relates to single-cell analysis and drug discovery. We're planning to launch this September. We planned already 6 one-hour modules, and we're going to have online office hours from the team members, and we're going to have cloud computing available. So what are the next steps? Jay's lab has done a lot of the transcriptomics, and we're working with them to make predictions using our platform isosceles. Kebon's lab is producing the nanoparticles. Our next step, obviously, is to put the compounds in nanoparticles and then to proceed and test these in the animal models in later years. We're thinking broad. Alzheimer's was mentioned in a previous talk. This platform is amenable to be used in different neurological diseases. So if you have a disease signature in Alzheimer's from AB secreting cells, or in the case of MS, B cells, you can try to reverse the disease signature using the small molecule approach that we have. So basically, we would have small molecules predicted to reverse the disease signature of specific kinds of cells, not all cells, but just the disease cells. In collaboration with Kebon, we can actually put the small molecules into the nanoparticles and target the disease cells to reduce either plaques or, in the case of MS, cytokines. So this has really been a wonderful team effort, really integrated. We meet every 2 weeks online with Jade Lee's lab and Kebon's lab and my lab. We have a program manager, Anna, and we have a bioinformatician, Rashika, and a lead, Rob Suter, as well. We've met already in DC last year, and we've planned our next meeting in Miami in the winter in January as well. We have a Slack group, and I think I've learned quite a lot, and it's a full circle, meaning that work from my lab in glioblastoma has influenced spinal cord injury, but it's going back, and I'm learning as well. So thanks.
Thank you very much. Dr. Ayad, there is a question for you in the chat, but if you wouldn't mind, for just the time constraints, if you could potentially just answer it in typing the answer.
Sure.
Thank you. Okay. We have our last project presentation, and then after that, we'll just take a brief break, just an adjustment on the schedule. We'll start the next part of the session with Liz Allen at 2:40 approximately. But our next presentation, we'll go ahead and get started. Dr. Laguerre-Turenne, Drs. Emerson and Blaney, and the title of this presentation or project is Targeting Dysregulated RNA Splicing in Neurodegenerative Diseases. Thank you.
Good afternoon, everyone. Can you hear me and see the slides?
Yep.
Great. So it's really my pleasure to represent the team here today and for this combined project, which is in close collaboration with Paul Blaney at the Broad and Michael Ward at the NIH NINDS. And So the goal of our project is to identify cell type specific splicing alteration and really use genetic screens to uncover modulators of RNA misprocessing. And for that, we are really leveraging complementary expertise from our labs, as I said, with Paul Blaney at the Broad and Michael Ward at NIH. And here is the most important slide with really the postdoc and students leading various aspects of this project in our 3 groups. We also actually have a lot of help from Robert Majowski, who is a science advisor, and is really helping coordinating the different efforts. And our bi-weekly meetings are now actually joined by Dr. Aziz Al-Kafaji and Brian Haas, who are at the Broad Institute and really expert in long-read sequencing and are helping a lot with bioinformatic analysis. And so really the project stems from the recognition that RNA processing is highly disrupted in several neurodegenerative diseases, in particular with multiple RNA binding proteins that are dysfunctional, either because their expression is reduced or because they abnormally aggregate with their ability to really regulate RNA processing, and in particular RNA splicing, being lost in various diseases, in particular neurodegenerative diseases. And so to canonical RNA binding protein that undergo this misregulation are TDP43 and FUS, which are normally located in the nuclei, and in patients, they are abnormally aggregated. And so these are disrupted in multiple neurodegenerative diseases, but very much a large proportion of patients with amyotrophic lateral sclerosis and frontotemporal dementia, but also for TDP43 in up to 50% of patients with Alzheimer's disease. And so we and others have really used various transcriptomic approaches as they were coming along to identify the normal role of these RNA binding proteins, but also to determine what are the splicing alterations that occur when these proteins are lost. And here, for example, for TDP43, we found multiple RNA splicing with different types of RNA splicing that can be altered in disease. And interestingly, in 2015, there was a group of Phil Wang at Hopkins described an interesting way that TDP43 is regulating splicing, which is by inhibiting the insertion of what we call cryptic exons, which are normally intronic regions, but they can be abnormally inserted in the major transcript. And so both Michael Ward and myself have led some studies that have really characterized some of these splicing events, and in particular in STATMIN2 and in UNC13, which are two major targets of TDP43, for which there are important efforts now to rescue these splicing alterations as therapeutic approaches. However, we really have a little understanding of many things in terms of splicing. First of all, we don't really understand cell type specific splicing events. Most of the studies have been done in neurons or neuronal-like cells or bulk tissues. And we also relied until now on short read sequencing and really don't have a good view of the diversity of the different transcript isoforms. And so for this reason, we proposed in this project first to define cell type specific splicing alterations that are associated with disruption of these RNA binding proteins. And for this, we will use isogenic iPS-derived lines. We are going to also determine splicing profiles in postmortem tissues from patients with distinct forms of ALS and frontotemporal dementia. And the role is to have to be able, first of all, to have a map of different splicing alteration in different cell types, but also then to select some of these most prominent splicing alterations and to use them as readout for the function of the RNA binding proteins, and to be able to do genetic screens to identify modulators of these splicing alterations that could represent new therapeutic targets in neurodegenerative diseases. And then in the last time, we will validate the functional impact of the identified modulators from the screens. So, as I said, we are really taking advantage of complementary expertise, and in particular of emerging techniques. So one is long-read sequencing. And so now it's heavily used, I think, by many in the field, and it's commercialized by PagBio, but it was actually developed in Paul Blenet's group at the Broad Institute, and in particular by Aziz Al-Khari, who I've mentioned is now very much integrated to this project. And so this long read sequencing really allow a much better characterization of the different splice isoforms that we could do until now with short read sequencing. We are also taking advantage of major resource which comes from the INDI consortium, which is co-led by Michael Ward, and which really provides the field with different iPS lines that are isogenic, in which various mutations have been CRISPR edited, and in particular mutations in these RNA binding proteins. We have also developed various methods to differentiate these iPS lines in different cellular models, different types of neurons, as well as glial cells and skeletal mesobs. And so we propose to do this long-read sequencing in these various iPSC-derived models. Then, as I said, what we want to do is to use selected splicing alterations to do genetic screens. And we are going to do in parallel two types of screens. One is a FACS-based genetic screens, where we use as a readout splicing. And for that, we use basically HCR fish, which is a way to really label abnormal splicing events. And here I'm taking the example of StatMin2. I've mentioned that there's a cryptic exon, and indeed with this fish, we can detect the cryptic exon in the iPS-derived neurons when we knock down TDP43. And so James Albroth in Michael Ward's group has really spent a lot of time to set up this flow cytometry protocol to be able to do large-scale genome screens based on splicing as a readout. In Paul Bleney's group, he's actually really at the forefront of developing screens that are based on imaging phenotyping. And so this is called optical pool screens. It was published a few years ago by his group, and contrary to other types of screens, it really allows to use as readouts image phenotypes. So any microscopic phenotypes that you can imagine, including fish or immunofluorescence, can be used with the cells being then transduced with various guide RNAs, so pooled libraries, and in situ sequencing, allowing for each individual cell to determine what was the perturbation. And so my group has been working very closely with Paul's group to really adapt this method to iPS-derived neurons, and we have made a major progress in this direction. So as I said, it's a highly integrated project between the three groups, and with actually efforts ongoing for all the aims, at least the first three aims. We have made major advance in doing long read sequencing in iPS neurons, as well as in postmortem tissues, and progress also in developing the methods for this pooled screen in neurons, as well as HCR fish facts-based screen. So we meet every 2 weeks as a team, but actually, I have to say the members of the team are, I think, interacting really daily. And people from my lab, many of them are actually doing experiments in Paul's group, and some students and postdocs from Michael went to Paul's group to learn how to do OPS, and people from my group went to Michael Ward's to learn several of the neuronal differentiation. So it's a very close group, and we enjoy very much this collaboration. So thank you so much for the opportunity, and let me know if you have any questions.
Thank you very much. If anybody has any questions for Dr. Liguritorian or her team, if you could please put them in the chat. Otherwise, we're just going to take a very brief couple-minute break, and I have 2:38 right now, and at 2:40, we'll go ahead and move on to the final session of the day, or section of the day, where we'll have 3 guest speakers, kind of zooming out and kind of looking at team science from more of a meta level, and they'll describe their perspectives and experiences. So take a couple-minute break, and then we'll rejoin with Liz Allen. Liz, are you ready? While Liz pulls up her presentation, just a couple housekeeping items. First, I want to thank our Combine awardees for taking time out of managing these very complex projects to come and do this early dissemination activity with us. The other thing is for anybody that's on the call that is interested in applying to the Combine program, I just wanted to point out something. The presentations that you saw today came into the previous version of the Combine NOFO, which had multiple aims. So you would have seen multiple aims in some of those projects. The current NOFO does not have multiple aims, as Karen discussed. So just so there's no confusion about like, why did this project get funded with multiple aims? It's because we revised the NOFO after these projects were awarded. So. Okay, with that, I am very, very excited to introduce Liz Allen. We recommend the contributor role taxonomy to a lot of our grantees, and I'm probably going to pronounce it wrong, but I guess we'll all learn together today. But Liz was, one of the people that developed the CRT as well, I believe, is it? I say ORCID, maybe it's ORCID, I don't know, but I'll let you educate us today, Liz. But so thank you very much for joining us all the way from the UK. It's the evening there, so we very much appreciate it and look forward to your presentation.
No problem. And can you hear me first?
Yeah, you are in desktop mode rather than presenter mode.
But you can actually see the slides, can you?
Okay, yep, we're good. We have your speaking notes, maybe switch to the display.
Oh yeah, hang on. Okay, hang on a minute, let me work out how to do that.
It's in the top right middle display settings.
Okay.
Yep.
And while we're getting started, if Clotilde and team could please answer the question in chat. Thank you.
So can you just clarify you can see the full screen right now?
You're good to go. Yep.
Brilliant. Thank you. Yeah, thank you very much for inviting me. Yeah, I'm calling you here from London, so it's great to be part of this meeting and really great to hear a bit of the multidisciplinary work. I just jumped in on the last two presentations, so really great to hear some of the use cases for actually this contributor role taxonomy. So I've got quite a lot of slides. I'm going to leave them with you, so I will share them with Corey, and I'm going to try and pick out the key points, but I wanted to really introduce you to what the contributor role taxonomy is about, where it came from, and how it's being used to support team science specifically and provide visibility to the roles that happen within a big team. So that's my credentials. I'm actually about to leave Taylor and Francis. I work at a publisher at the moment, but I also have a visiting fellowship at the Policy Institute at King's in London, and I used to work at Wellcome as well, so I've got a funding background too. So just a quick summary. This is what I'm going to talk to as quickly as I can, but hopefully in a way that makes it easy for you to kind of grab hold of it. Why visibility of research contributions matter. Many of you probably already know this, and many of you have probably come across credit, particularly if the Institute is actually recommending people be aware of it and use it. I want to tell you a little bit about where it came from, and I wanted to show you some of the values of using contribution information. This is all probably pretty obvious to groups of really senior and mixed ranges of all different levels of researchers. So why are research contributions important? They support careers, it's important for research culture, it's important for research assessment, it's important to know who's contributing to the different types of research that we do and what kind of roles are important for recruitment, to bring efficiencies, and just to be able to know what's going on in research for trust and transparency too. Increasingly, we're seeing the reason that some of the rationale for using research contributions in research for research accountability and integrity as well, which is becoming, particularly in the publishing world, a really big issue at the moment. And I'll explain a little bit about that in a moment. But essentially, it's about providing more information about the contributions and the different kinds of roles that exist within science and how researchers are contributing. We kind of know that science is a big team effort, and necessarily so. And we just need to encourage and value this as being core to what we need within an effective research system and the culture that we work within. And there are a number of initiatives out there at the moment designed to help support a more holistic and representative view of the different types of science and how teams work. You've probably all come across DORA, the Declaration on Research Assessment. And one of the key recommendations and statements within DORA is in the red there, that outputs from scientific research are many and varied, And we need to recognize those, not just focus on where someone published and whether they're first author on a paper. And the same for QARA, which is the Coalition for Advanced Research Assessment. This has particularly come out pretty strongly within the European context. But again, it says the same kinds of things, that it's really important that we recognize the diversity of outputs. And so we need to find ways to make sure that we can do that. And that's really where CREDIT came about. So CREDIT, CRT, stands for the Contributor Role Taxonomy. And initially, it's been focused very much on published work because this is the most easy and most visible manifestation of research output. So we've been looking at providing more information on research outputs that are published beyond just focusing on author names, particularly because authorship increasingly, and many of you know this, it doesn't reflect the range of or nature of the contributions to research. And typically author information and contributor information is written or hidden in an acknowledgement section or not very well described. And people use proxies about author position for all kinds of interesting reasons. And if you just take a moment, if you haven't seen this before, this is quite old, but it still applies. We hear it a lot from researchers that this is still a challenge. If you read some of these, I mean, it's kind of a humorous look, but it just shows there's lots of game playings within authorship lists and lots of challenges with traditional being able to derive information from those kinds of authorship lists. And it doesn't mean that authorship lists aren't important. Of course they are, and criteria to be an author is absolutely important, and that's enshrined in most journal policies. But what it doesn't tell you is what kinds of things these different authors did, and particularly what some of the more middle authors and the range of different authors within this did. And we can do a lot better because we can capture that information much more easily. Another thing that CREDIT came out was we see a particular demise of like lone author and small author list papers, and particular trends to team science in many disciplines. So again, I'll share these slides with you, but there's so much research now showing, particularly in STEM subjects, and particularly in medical and clinical research, and much bioinformatics and physics areas too, that the share of research authored by one person or a small number of people is declining, and it's declining pretty steadily over time, and that's all over the world. And then this is like an extreme example, but this is kind of, you see examples of this now, consortia authors. We have lots of work coming out of the physics community. Perhaps this is a great example from CERN in Geneva, with over like nearly 3,000 authors from 169 different research institutions. And how do you use that information in terms of trying to make sense of who did what and what valuable contributions people make? So it's kind of a bit of a deterrent for a lot of people to kind of put their name to some of these papers as well, because you just get hidden within a huge range of authorship. And it's really important for researchers to show what they did. Research assessment challenges and demands have increased, and in many ways, rightly so. But instead of just focusing on being first author on a paper, there are lots of other ways that you could acknowledge the importance and the roles that you presented within research. I just wanted to briefly mention, this is something that it would be great to talk to the institutes and lots of funders a bit more about, but publishers are increasingly facing with lots of issues with provenance and research integrity, particularly from certain parts of the world, but it is all over the place. And we need to keep making sure that we as publishers are assuring the trust in the content that we publish. There's a whole range of issues around research integrity at the moment in terms of fake papers, fake data, data manipulation. Many of these are intentional, but some of them are actually unintentional issues that are emerging throughout the whole system. People citing news that research that's perhaps been retracted, pressure to publish, driving potential papers that don't really have the peer review criteria and assurances around them, lots of kind of issues. But what it means for a publisher is that we need to be more and more on our toes around who and what are responsible for the content that we publish, because publishers are liable for much of that content and need to be making sure that we have provenance data absolutely pinned down. And that's just not authorship, it could be all other things, in which case contributions is really important too. So if there's a problem with the data, we know who's responsible for the data. And mainly to contact, and so we can do something about it. Often research integrity issues are unintentional, and we're happy to work with researchers, but it is important that we have lines of dialogue open to make sure that content and research is trusted. And then more practically, it's really useful to have information about what authors did, and some of the pressures of publishing in a print world, which doesn't exist anymore, means that we can capture information and share it more easily within the published record. So just briefly, this is the contributor role taxonomy. It was developed following, I was working at Wellcome at the time, and I was working with some colleagues at Harvard who wanted to provide more visibility around the contributions that are being made and captured on research article papers. That's where we started. So we looked at what journals were existing journals were already capturing around contributor roles. We focused very much on STM to start with, particularly because this is where the challenges were being raised. We tried to look at, if we did something, we needed to keep it simple, because we know publishing a paper is complicated. It's as complicated as applying for a grant. I don't know whether it's quite as complicated, but there are loads of different ways of publishing, and we don't want to introduce contribution issues and challenges and more unintended consequences for having a taxonomy that when you publish, you have to add extra information and it takes a load of time. We don't want to do that. But having known that people are already doing that in a different part of the paper, we just wanted to try and streamline it somewhere else and to be able to use the data better. We pilot tested in 2014-15 with PLOS. So the Public Library of Science journals switched off the existing 5-role taxonomy they had, and they replaced it with credit, which is a 14-role taxonomy. It became approved as an ANSI NISO standard, so publishers are being advised to use this standard from 2021. And I've been part of a small group of people. We've been awarded funding to help continue drive the concept and adoption. And one of those things, it's great to be able to talk to you guys, but we have some funding from the Sloan Foundation and Wellcome to continue driving that. And again, I mentioned I'll share this deck, but there's some links if you wanted to find out more. If you've not come across it, again, I'll share this. I won't go through this now, but this is the 14-role taxonomy. The roles are down the left. So publishers, when you submit to publishers using credit, they ask you for all the authors that you're submitting, which of these roles did you do? You can have more than one. The paper doesn't have to have any of these on. It can have, obviously, there'd always be a writing original draft and probably reviewing, but the other roles are interchangeable. You can have multiple authors writing and contributing to some of them. You can have just one author doing them, or the roles might not be relevant. But each of them are accompanied by a definition around what that means. Lots of publishers are now using credit in their submission systems. So just some examples here. So I suspect quite a lot of you in the audience, it'd be great to know, have come across this. And this is just an example. I pulled this paper today from the Royal Society Open Science Journal today. This is how it manifests typically, although it varies in journals. This is how you can see the information, and it's captured as part of the metadata. So there are 5 authors on this paper. The first author is Nikki Cole. And this is, if you click on the XML on the HTML on the website of this journal, you click on the author list and it brings up this information for Nikki Cole. So you can see her ORCID ID. So Nikki also has an ORCID ID, which means she has a researcher ID. All papers are linked to her through the sort of Crossref ecosystem. So when you publish, you're given the option to link them to an ORCID ID. So if you haven't got an ORCID ID, please do sign up. I don't know if it's being encouraged at NIH, but quite a few funders are encouraging the use of this now. And ORCID is a broader system to link researchers to their research output. And if you can see the statement at the bottom, it says what she's contributed. And you can see the different roles. Of those 14, she's selected that she's related to. And I've highlighted in red, there's 3 that are unique to her, actually, of those 5 authors. And then if you look at Eva, who's the second author, she's done different things. This time, uniquely, the software work she's worked on. So you can see that you can actually start to get metadata linked to an author through an ORCID ID that she's been responsible, or either of those authors have been responsible for. And that's how it works across a lot of publishers. So when you submit an article, you're asked to complete the contributor role taxonomy according to who did what. And just before I finish, I wanted to show you some of the value in credit, and we're seeing more and more of this. The key thing is that metadata is now starting to be captured across systems. Hopefully, we're hoping to talk to PubMed. We're talking to Crossref to make sure that that metadata that publishers are capturing can be surfaced through systems that are bibliographic indexing databases. So not only can you find authors, you could look for someone who's a real specialist in data. you could look for someone who's a specialist in software, and we're hoping that that's going to hopefully pan through the whole system. We'll also be great for looking for peer reviewers, and I know that some of the publishers are looking at that now, so you could target specific reviewers. And again, funding agencies could have a use for that. So DORA have acknowledged that credit is a really useful thing. It isn't perfect, it doesn't capture every role in the world, and we're looking to evolve it over time as any taxonomy. But you have to be careful around taxonomies to not keep changing them through it's instead of step change, you kind of need to make sure you're ready to change them before you change them. But it's recognized in DORA as a way to recognize different contributions and value other kinds of outputs. And then specifically to team science, this is actually a few years old now, but they've issued an update in 2020, which is also a few years old. But the UK Academy of Medical Sciences published a review very much talking about some of the issues that I think you guys have been talking about today, is helping to incentivize people to get involved in team science. And they included a section on recommending that publishers provide open, transparent, and structured information around contributions, therefore using credit, which is what many publishers are now starting to do. And more information around the inputs to that as well. So funding information is the other thing that publishers are trying to capture more systematically. And that's a big topic. That would be great to talk to you about that too. But funding information has traditionally been hard to capture in a way that's useful to funders through publications. And really importantly, in ways that don't add administrative burden to researchers, because there are so many burdens on researchers now, particularly in the whole open science space. And sharing your data, sharing information. We just need to make things really simple, but it's still important if we can do that. And then finally, there are a number of studies now coming out of the work that credit and contributor roles shows in the literature. So there are studies showing the difference, labour and evolution of roles. So looking at what do middle authors do? Is there a division of labour in terms of the types of things that they do? Are they responsible for data? Are they responsible for software? And looking at some of the things around that. There's some gender studies underway as well, looking at author lists, what the actual contributions of different authors do according to gender, which is really interesting. And these all have policy implications, of course, so it's really interesting to see that too. Some work on politics of team science and whether people overestimate what they've actually done, and we're looking at that a little bit. And then particularly for publishers, we're looking at its role in helping us with research integrity issues. We're thinking authorship lists just don't give us that clarity of vision and the accountability for things that we're starting to see where there are problems in aspects of research as well. So it's a really good way to add extra accountability into what we're publishing so that we're making sure that there's trust in the content that we publish. And that's the core to a research publisher's role. Couple of unintended consequences. We need to be a bit careful about proportionality. We don't want to make it a metric that people seek. We never wanted to do that. We know there are enough issues around research metrics. But I think it's information that's kind of there already. All we've really done is surface information around contributions. Most people who are contributors would have been authors anyway. We're just providing extra granularity to that. We want to keep it simple. We're looking at across other disciplines. It seems to be flying, and there's a lot of adoption across in STM areas because of the nature of the contributor roles, but we obviously have feedback from other areas that we haven't included roles that are important in other humanities and social sciences, for example, but we've started somewhere. And then to keep the taxonomy up to date, which of course relates to that too. So I'll stop there. I'm aware I didn't have a lot of time. I could probably spend lots of time talking to you about this. But yeah, it'd be great to get your feedback. If you want to find out more, look at the Credit NISO website. It's actually hosted by NISO. We're looking to put some updates on there now. And I'm on LinkedIn if you want to just contact me as well. I'm about to change roles, which is why I haven't left my TNF email around. But if you do want to contact me, Corey's got all my details. And yes, there I am on LinkedIn if you want to contact me. So I'll stop there. I hope that was useful.
Thank you so much, Liz. I know, at least speaking for Karen and I, we'd love to have you come back and talk for hours. You know, this is a fascinating topic that touches, you know, really every single person on this call or anybody, you know, developing scientific products. I just want to add one note. I know Karen's going to have questions. This is a topic she's very passionate about. But I just want to say, you know, like, kind of the purpose of the seminar, I think Liz's talk kind of embodies, the idea of building this community of practice. Karen and I refer our applicants and awardees to the CRT, and we were coming up with speakers for this. Karen came up, she's like, well, why don't we see who's working on CRT? And then we reached out to Liz, and not only, of course, as you have seen today, Liz is a wealth of information, experience, and credit assignment, but she's actually been working on a lot of things that Karen and I have been also working on. And so we're able to kind of, remove ourselves from these silos. And now we're hoping to collaborate and work together with the National Library of Medicine and PubMed to try to integrate some of these considerations that Liz talked about in the context of STEAM science. So, really, this is what we're hoping that comes from these talks as we do these seminars, that people can connect, find collaborators, and can continue to drive this field forward. So I'll hand it off to Karen, but thank you, Liz.
Yeah, Liz, this is probably a half-formed question, and you may have touched on this, but I'm curious about these keywords, the contributor roles, because I would imagine, depending on the field, then you would have specialized type of roles. So how to manage that in a way that you have just a sufficient set of keywords, but at the same time, building in flexibility depending on the needs of the field?
Yeah, that's a brilliant question. Almost like, I don't know if any of you have come across this issue before. It's like a sort of minimum viable product, right? We started with a really basic, simple set. Some publishers, what they do with there's an explanation associated with those 14 roles, and the explanation, the publishers, the way that the publishers have implemented, they can add a bit more explanation into that roles and say, well, we mean these kinds of things, we mean these kinds of things. So data curation, or we called it data curation. I appreciate that probably doesn't cover a lot of the kind of data that some of you guys on this call are doing, but it's meant to mean the data specialist, the person who dealt with a lot of the data on that call, and that varies according to discipline. It's really hard to get it to fit every single discipline. And we know there's more work to do, but we hope we've made a step with at least getting it starting to be used. We've also got some technical, a lot of it is to do with keeping the metadata underlying it very simple, the way that publishing metadata, so all the author details, all the funding data, all the institution details, all the content, it has to be fairly simple. So we needed to keep it simple too, but really good question. And I think what we're looking at, since we developed it, I think science moves so fast, particularly in the whole data space, and software and visualization. AI, everyone mentions AI or whatever that is, technology, any kind of things really changed that space. And so we are aware that we might need to develop more specialized kind of layers below that, but we wanted to keep it simple in the first instance. Really good question. And it would be fantastic to potentially work with you guys to look at that and how it could be evolved in certain areas too.
Thank you.
Thank you, Liz. Yeah. Okay, Liz, there is a couple of questions in the chat. If you wouldn't mind just replying to them via typing, we would appreciate that. And then we'll go ahead and move to our next talk, which is by Alyssa Pacchini-Schaefer from the Simons Foundation. And the title of the discussion is Facilitating Collaborations in Basic Neuroscience at the Simons Foundation.
Hi, thank you very much, Corey. Let me take a second and share my screen here. Let's see. Oh, you probably see something else right now. Give me 1 second. All right, how's that?
Looks good.
Thank you. All right. Thank you, Corey and Karen, very much for the invitation to speak today. My name is Alyssa Pacchini-Schaefer, and I am the Vice President and Senior Science for the Neuroscience Collaborations at the Simons Foundation. And Corey and Karen asked me to speak about facilitating collaborations in basic neuroscience and some of the things that we're doing here. So I first wanted to start out with a little bit of information about who we are at the Simons Foundation. The Simons Foundation is a philanthropic organization dedicated to advancing the frontiers of basic science. And we do this through grant making, in-house research, and public engagement. Since its founding in 1994 by Jim and Marilyn Simons, we've really been champions of basic science. We believe in asking big questions and providing sustained support to researchers working to unravel the mysteries of the universe. And we believe the brain is one of the mysteries of the universe, to put it bluntly. The Simons Foundation makes grants in 4 areas. So we are much broader than just basic and fundamental neuroscience. The 4 areas you can see here on the left, you can think of this sort of as our extramural programs: mathematics and physical sciences, life sciences, autism and neuroscience, which is the division that I'm in, and science, society, and culture, which used to be called outreach and education. Our Flatiron Institute, which is really our intramural research program, it was opened in 2016 and conducts computational research. And there are 5 centers: computational astrophysics, biology, mathematics, neuroscience, quantum physics, and they are all supported by a scientific computing core. So back to neuroscience. There is neuroscience going on at 3 parts of the Simons Foundation. One is in our in-house research, the Center for Computational Neuroscience. Under Science, Society, and Culture, we have an editorially independent publication called The Transmitter, which communicates about discoveries in neuroscience research. And I suggest that you guys go check it out, thetransmitter.org. And then what I'm going to talk about today mostly is the Autism and Neuroscience Division. Under the umbrella of the Autism and Neuroscience Division, we have our neuroscience collaborations, which is most of what I'm going to speak about. And we also have the Simons Foundation Autism Research Initiative. And within our Autism Research Initiative, we do have a few collaborative projects. So the mission of the neuroscience collaborations at the Simons Foundation is to support and convene scientists to address fundamental questions about brain function. We work closely with the Simons Foundation Autism Research Initiative, or SAFARI. And so the mission of SAFARI is to advance basic science research on autism and related neurodevelopmental disorders. So one important thing is that each neuroscience collaboration is 10 years, they're time limited, whereas SAFARI, the Autism Research Initiative, is a permanent initiative of the foundation and is going to be around in perpetuity as long as the Simons Foundation exists. I'll start and give you a little bit of background about the Autism Research Initiative before I dig into our science collaborations. So the activities of SAFARI can be categorized into 3 buckets: convenings, which are workshops and meetings; grant making, which they do in individual ways, direct research awards; more collaborative, the collaborations. We have early career awards and some institutional giving. And we also generate resources in terms of biorepositories, cohorts, and data. So one of SAFARI's greatest impact since its founding has been on the genetics of autism. So back in 2007, we knew that autism was one of the most heritable neuropsychiatric disorders, where between 70 and 90% of identical twins both had autism, about 31% of fraternal twins both have autism, and about 20% of siblings both have autism. So from those numbers, you can tell, that the risk is both genetic and environmental. And so in order to investigate the basic science underlying this, the SAFARI started by making large investments in human genetic studies. And you can see here, we started with the Simons Simplex collection. We are still running an autism inpatient collection cohort. We have a Simons, what we call Simons Searchlight, which is a genes first cohort. So you are brought in through a genetic diagnosis of a neurodevelopmental disorder. And then we have Spark, which is a phenotype first cohort, where if you have a diagnosis of autism, we are then collecting genetic information. So, and through this work, SAFARI has made major progress on the genetics of autism. Spark alone has identified 167 genes and 43 copy number variants that are significantly associated with autism. And in addition to the cohorts that I just talked about, SAFARI also does neuroscience research on the basic science underlying autism and provides resources for this scientific investigation, including there is a postmortem brain bank, iPS cell models, mouse models, rat models, zebrafish models. And through Safari Gene, there's data analysis tools, there's research mats and clinical research associates. So if anybody is interested in these resources, they're open and free to the community, and there are ways to request them through our website. And so I suggest you go to safari.org and check those out. So genes are just part of the picture to truly understand autism, and Safari is funding research at other levels, from networks to circuits to behavior. And two of the ways they are doing that is through their larger scale collaborations. One is called the Autism RAC Consortium, and the other is called Sex Differences in Autism. And I will get into a little bit more in just a minute about how we structure those collaborations and how we think about collaborative team science. So the collaborations were really the brainchild of Jim Simons, and I just want to take a moment to acknowledge his life and legacy here, as he passed away last month. He believed that if people worked together deeply, the whole would be better than the sum of its parts. And this was born out of his experiences in mathematics and running the math department at Stony Brook, among some of his other many career accomplishments. So he empowered us at the Simons Foundation to set up and facilitate collaborations. And I work on the neuroscience collaborations. And though that's what I'm going to talk about today, because that's what I do, you guys should know that we also have collaborations in mathematics, physics, and the life sciences too, in addition to autism and neuroscience. And Jim set the parameters of these. The collaborations are time limited. They are to be 10 years long, and that is long enough for deep investments and progress around a central topic, but time limited to allow for the evolution of new ideas and for us to evolve the things that we're working on and investing in deeply over time. So in neuroscience, there are currently two collaborations running. One is the Simons collaboration on the global brain, and the other is the Simons collaboration on plasticity in the aging brain. So the global brain has a lofty scientific goal. It aims to expand our understanding of the role of internal brain processes in the arc from sensation to action, thereby discovering the nature, role, and mechanisms of the neural activity that produces cognition. And the focus here is really on neural coding and dynamics at single cell resolution and the computational and data analysis approaches that are needed to support that. And the reason that the goal is this and the level of sort of understanding is at the single cell resolution level is because this is what Jim believed, and he was convinced would help reach the goal of funding this program, which was to understand how a thought happened. That's why Jim used to say he wanted to fund the global brain, was because he wanted to understand how a thought happened. So the Global Brain collaboration was started in July 2014. It was set to sunset in July 2024. However, with COVID, we gave them a year extension. And it funds about 96 investigators across almost 30 sub-projects that have an average size of about 5 to 7 investigators. But you will hear about one outlier project, I believe, right after my talk, because I think Hannah Bayer is going to tell you about the International Brain Lab, which is one of the it's the largest collaborative project within the global brain that we fund. And you can see here on the right, I like to call this the SCGB Connectome. It is the web of collaborative interactions, either by co-publications or by co-grants that we give of all of the investigators in the Global Brain Project. And this little, this is actually an interactive graphic on the website. If you go to our webpage, you can click on this, and then every time you click on one of the investigators, it'll show you what publications or what awards that they have together. So it'll give you the data on how all of these things were, how that graph was made. The other neuroscience collaboration that is currently running is the Simons collaboration on plasticity in the aging brain. And the goal of this project is to discover the mechanisms of resilience and maintenance in the aging brain, and to establish a baseline for age-related changes in plasticity across many model systems. So the aging brain began in March 2020. I actually got the go-ahead to start this collaboration March 5th, and then the world shut down. But and it is much smaller, and it got off to a great start, actually, in many ways, despite COVID. This project is funds 31 investigators across 7 smaller, more focused projects. And here, as in the Global RAIN, we facilitate collaboration through data sharing, workshops, annual meetings, building pipelines, and encouraging preprints and many other things. And I will get into the details of all of this in just a minute. And importantly, with this collaboration, at the very outset, we initiated and organized, facilitated and curated collaborative workshops to align on time points, protocols, data modalities, data outputs across all of these projects with an eye toward direct comparison. So we act as the the scientific program staff here at the foundation, we sort of actively facilitated the workshops and the conversations to have our investigators align on all of these things to achieve this goal. And the whole point of it was to create a database, an aging brain database. And so what you're looking at right here is an infographic on how all of these projects align and the comparisons that we're going to be able to make over time as the researchers generate data and deposit it into this database. So that is something that we're actively working on, and these things are going to be coming online. The data is first going to be shared within the collaboration, and then this database in the next, well, I'm not going to commit to a firm timeline right now, but the database will be shared with the public and available to anyone in the neuroscience research community to use and access the data. And so right now, we are in the middle of a process to identify the next neuroscience collaborations at the Simons Foundation. So the next places that we are going to be investing money in fundamental and basic neuroscience research. And so what we did was we put out a call for vision statements for areas of neuroscience poised for high impact funding from the Simons Foundation. And we had a focus of areas that would bring together 2 fields that don't communicate or collaborate often, and that can make a big impact. That significant progress could be made toward understanding over 10 years, and significant progress could be made toward understanding in the next 10 years. We received 251 vision statements, and we narrowed that down to 20 that we requested full proposals from. And you can see here the sort of large categories of areas that we asked for full proposals from. There's sort of neuroimmune interactions, brain, body, interoception, social brain, social communication, omics and computation, learning and generalization, and learning and development. Now, in this call, we didn't directly specify, but we gave guidance that we thought from our experience running a very large collaboration and a smaller one, around 20 to 30 PIs seems more optimal from our experience to focus on, have a focused goal. And having those 20 to 30 PIs organized into smaller groups with more clearly defined scientific pieces and goals also is one of the ways that we feel like these collaborations can make significant progress. We also have found that deep collaboration is possible with larger groups if you invest significant time and resources into the collaborative infrastructure. And I think Hannah's going to tell you about that in the next presentation about the International Brain Lab. So what are the things that we're thinking about when we are selecting the next neuroscience collaborations in terms of collaboration? And I think one of the things that we really look deeply at is that we prioritize not only the science, but as highly we prioritize the interpersonal parts of the collaborative process. So collaborations and deep scientific collaboration is really built on trust. And there are many things that go into trust, but part of it is people knowing each other well, people being able to communicate well, and agreements and transparency that everyone knows what to expect and rules that everyone lives by. And these sort of bolster and facilitate the trust that leads to deep scientific collaboration. So during the application process, we get written applications, but we also do sort of, we call them like virtual site visits, Zoom calls, where we look for interpersonal dynamics during the application process, evidence that teams are thinking together and that there's a dialogue and discussion amongst the investigators and a give and take. We ask for detailed leadership, communication, and decision-making plans. And we also make sure that it's not just the PIs that are involved in this, that all of this is communicated and agreed upon by the trainees as well in the collaboration. These detailed plans, we ask for things including transparency in decision-making, transparency in author assignment, which is very pertinent to the previous talk, transparency in the financial resources and decision-making, how you're going to communicate, and evidence that they are putting time and resources into these things, into the interpersonal parts of the collaborative project, because resources, i.e. money, and time commitment for those things we found is as important as resources into the science in order for the scientific project to succeed. We also ask for a commitment to data sharing. And we look at this deeply when we're evaluating what these projects are. And this goes beyond sort of formatting your data after you've collected it and depositing it into a public repository. I mean, that is important, but it's not enough to have the data just dropped into a public repository if we want the data to be used and analyzed in the future. We've found that you really have to fund people to facilitate, support the data, support the pipeline and sharing, to advertise what's in the data, to advertise what's out there and get the information out there to others, and guide people through the data and the pipelines in order for sharing to be meaningful and data to not just sit there. And so software engineers are especially important in this, but so are sort of project managers. And because it takes people to access the data well in meaningful ways where it can be analyzed and used by another group. One of the other things we've been thinking about is that, some of the feedback we're getting is that, theory groups or others would love to actually go in and use other people's carefully collected and curated data sets. But that takes money too. You need to pay people to do those things. And so again, after my talk, you'll hear from Hannah Bayer about how we've been talking to some other groups about potentially funding RFAs to use carefully curated and supported data sets for new discoveries so that people can be paid to actually think about scientific questions. And it's a way to harness all of the information and knowledge that is in those data sets in ways that couldn't have been done previously. Okay. Another thing that we think about when we're evaluating for collaborative projects is connections and communications, not only on the level of the PIs, but also on the level of the trainees, and making sure the trainees know about the high-level goals of the awards. Who else on these collaborative projects are working on things relevant to their own work? We organize trainee-only meetings in addition to PI-only level meetings and all-level meetings in order to make sure that the relevant information is available and exchanged among all different levels of people working on the projects. And we do this, a lot of it because our collaborations are distributed is virtual, but we also are really, one of the things that we have found that builds the trust to make a collaboration successful are in-person interactions. Some of those are formal, like annual meetings where we present the results of our work. But as important as the communication about the data and the work is, which is our utmost goal, we've also noticed that the meals, the breaks, the social times are as important to the collaboration and the scientific discourse and building trust that leads to real open communication about the work as the talks themselves. So we provide resources for in-person meetings, both formal scientific meetings, like I just mentioned, and even smaller, more informal meetings, exchange of trainees going to another lab to learn a technique for a week, trainee-only in-person meetings where they get together and share data and think together about how the work fits together and how they can help each other and collaborate. So on all different levels, we found that that's really, really fruitful. Another thing that we that we do at the Science Foundation is that our scientific staff take an active role in convening and ensuring collaboration. We make it clear that they know that the PIs in our collaborations know that we are their allies in the success of the project. We are there to brainstorm, think through roadblocks. We have principles of flexibility with our funds to help to pursue new directions within the scope of the work, as long as we're in conversation and they're discussing it with us. And it's our job to understand how all of the science is going in on these projects and how it fits together. And so we oversee the scientific goals, we understand the connections between collaborators, we work with the investigators to curate meetings and interactions to bring out things that they might not see from their small vantage point, because we have the bird's eye view. And finally, when we ask for progress reports, because to do our due diligence, we need to understand what's going on in these awards, we don't just ask for scientific progress. Of course, we do ask for that. It's very important to what we do. But we've noticed that when we ask for collaborative progress in our reports and our meetings, it changes how seriously they take the collaborative data sharing aspects. When we ask for updates on how often they meet, who's talking, how they're sharing their data, how it's getting out there, how they're communicating about that data, they understand that it's as important to us and it's important to the science as the publication that's going to come out in PLOS computational biology next week, right? And so those tracking progress on all of the milestones that are important to us is one of the other things that we have found has helped. And so I think with that, I could tell you a little bit more about the collaborations on the Center for Computational Neuroscience. That's a little bit outside of the work that I do specifically. So I think, and I'm pretty much at time, so I think I will end there, and thank you very much for having me.
Thank you very much, Alyssa. If you have questions for Alyssa, go ahead and put them in the chat. Thank you for the presentation and the critical advances that the Simons Foundation is making, not only in neuroscience, but as you mentioned, physics, math, and the life sciences. I think, earlier today when I introduced the session or the talk, the field of team science is still growing and we're still learning, but originally most of the research were focused on small teams, and now more recently it's moved to large teams, but very little work has been done thus far for funders and program design of team science. And so I think it's really critical that we collaborate with other, foundations like the Simons Foundation to understand, their best practices when it comes to designing and administering team science programs. So I think we still have a lot of work to do. And these are the first steps to kind of understanding how other people approach this and coming together and figure out what works for everybody. So thank you. I know there's a couple questions in the chat from Grace, and then
I can answer that.
Well.
I can answer that quickly verbally if you'd like.
Yeah.
So Grace asked, do we require authorship across the entire team versus subgroups of the team publishing their work? We don't require authorship across the entire team. I think maybe in some collaborative projects that would be necessary, and I'm sure Hannah will tell you about how they do authorship at the International Brain Laboratory. We let that be driven by the science. One of the things that we are pushing for the new collaborations that we are about to select is that we are going to require what it takes to get authorship and what authorship means. And some of the things that Liz was talking about earlier with the resources that she has developed, and just making those expectations transparent and clear when you are embarking on a project. I know one of the things that we've been really happy that the International Brain Lab has implemented is sort of a registration of who's working on what project, so everyone in the collaboration knows and there's no surprises about potential, sub-projects and authorship throughout the collaboration.
Great. Thank you, Alyssa. I really appreciate that, and also appreciate the emphasis on basic neuroscience, which is something we also emphasize at NINDS. And also I appreciate the emphasis too on team science as social activity and looking at what's going on in that aspect when you check in. So I'm curious, how often does your team check in with your project teams? And also, what are the usual roadblocks that you see when you check in?
Yeah, so we check in multiple different ways. We have an annual meeting every year where everyone comes in person to the foundation. We also have in the Aging Brain Project, we have online quarterly meetings where we check in as well. And so there's one in-person annual meeting, there's 3 online quarterly meetings, and then we also have a virtual site visit every year. So we're approximately checking in 2 to 3 times a year with each group in various different ways. And the problems are, I wish I could say that there is a usual problem. There is not a usual problem, right? I mean, people are all different. The science is all different. And it's really, individual to the projects and the people. But I will say that just knowing that there's a culture of us being engaged and that there's somewhere to go with problems is very, very, as I've been told, is very useful from the other side.
Right. And do you have some metrics of success?
Yeah, we're working on that. That's hard.
Yeah, right.
Oh, that's really hard. If anybody has any good ideas, please tell me. I don't know that we found a way to capture it exactly.
Very helpful. Thank you. And like what Corey said, we should all be talking about how we can better effectively support team science, as well as make sure that we help these teams be successful.
Thank you so much.
Thanks, Elisa.
Thanks, Lisa. Yeah, we have a ton of our applicants and investigators and NINDS are Safari contributors, so definitely a lot of overlap in mission. Okay, to take us home today, I'll introduce Hannah Bayer. Just, again, just more towards the kind of the value of this seminar series. When we were looking at speakers, Karen's like, oh, I think I have a good speaker at the International Brain Lab. I was like, okay, sounds good. And she sent me the information, and I had been already working with Hannah on through the Brain Initiative on a Delphi study to identify consensus for best practices for trainees that are in team science programs. So again, it's just about breaking down bridges and silos and getting people connected. And then I had a call with Alyssa, who you just heard from, to kind of talk about the conference. She was like, "Who are those speakers?" And I was like, I'm going through them. She's like, "Oh, Hannah's one of our awardees as well." So it just goes to show, sometimes you just got to set stuff up, like Alyssa said, get people in the same room and have conversations, because we're all kind of doing the same things and working like almost in circles around each other. And now we're all in the same room and can kind of have more impact. So it's really cool. And so I thank you both for joining us, and I'll leave it to you, Hannah. Thank you.
Super. Thanks so much, Corey. Thanks so much for inviting me, and to Karen as well, for giving me the nod. I feel like my talk has been really well queued up both by Liz and by Alyssa. And I want to say thank you. Obviously, the Simons Foundation has been incredibly generous, as has the NIH, in supporting the IBL, what is truly, I think, an experiment in team science. So I'm going to talk, I'm going to sort of range from sort of high level to mid level to sort of very operational detail. So people should ask questions, and feel free to ask me to develop things that whatever interests you. There's plenty I could talk about. Okay. So I want to start at the beginning. We have a lot of examples of successful labs run by 1 or 2 PIs, right? There are lots of ways that you can successfully run your lab. You guys are all successful PIs. You've been around the block. You know what works for you. But we don't have nearly as many models for how to go beyond that to scale up to a model for managing a research group, a much larger group. And so how do we know what are the best practices for this kind of team science? We have to do the science of team science, right? We have to go through this process and learn from experience. There's some theory we can use, but in large measure, we're going to learn from trial and error. We're going to try and do our best, and then we'll correct when we get it wrong. And so I think that will be a little bit of a theme in my talk, is that the IBL, we started out with sort of like a very clear vision of what we wanted to achieve, and we implemented something that we thought would work, and then we iteratively fixed that to try and get closer and closer to what we thought would make us a successful organization. So first, let me tell you a little bit about who we are. So we are a large distributed collaboration. We're a network of about 70 researchers, a mix of PIs, postdocs, graduate students, technicians, and staff. And we're a mix of theorists and experimentalists. We're in about 20 laboratories in 5 countries. But one thing that it makes us, I think, unique is that our main mission, which was to collect a brain-wide map of activity in a decision-making task, relied on a single species doing a single behavior using one data collection apparatus. Now, this is pretty unique, and it created some unusual constraints, but I think it also enabled us to create some infrastructure, or required us to create some infrastructure and some standardization that may be useful for everybody. So even though we're at the far end of the spectrum for neuroscience collaborations, I hope that what I'm going to tell you will be relevant to you, even if you're a group of 5 labs or fewer. But I think hopefully the lessons are pretty general. Okay, so just to say that we have a scientific goal, which I won't talk about today, which is about understanding neural circuits. And then we have a couple of sociological goals. We're really committed to sharing data, to open science, basically, to sharing data and infrastructure with the community. The thing I'm going to talk about today is that one of our goals was to change the way neuroscience is done by demonstrating how a large group can pool their efforts and work collaboratively. Okay, so I'm going to start with how we decided to organize ourselves. And again, I want to say we modeled ourselves after the Atlas collaboration at CERN, which maybe you saw in Liz's slide earlier, has hundreds and hundreds of people. We're not quite that large, but we thought that it might make sense for us as an organization to adopt this kind of flattened hierarchy that they had used. And I want to say that, like, for your 5 or 7 lab collaboration, you might not need something quite as big as this. In fact, we're organized not quite as extremely as CERN, but I want to point out sort of a few aspects of our organization that I think are part of what make it successful and might be usefully implemented even in a smaller team. So we have 3 sort of leadership bodies: the General Assembly, which is the voting body, and that's made up of PIs. Although there's also, you can see there's a researcher representative and a staff representative. Then we have an executive board, which manages day-to-day operations. But the real day-to-day work is actually going on in small teams, which we call working groups, where around specific specializations or milestones. For example, all the physiologists may meet in a group, or all the people working on a particular large-scale platform paper. And these groups are coordinated by a chair and an associate chair. And then the decisions and proposals that come out of these groups are shared back up to the executive board and the associate chair's committee. So it's a flattened hierarchy. That means that the information is basically flowing and decisions are made at multiple levels. We have working groups for streamlined daily operations, where you couldn't have the General Assembly or even the executive board sort of, weighing in on everything every day. But we want to make sure that information is coming from all different levels and being shared up to the leadership groups so that they can make the larger scale decisions effectively. And again, I really want to point out the importance of trainees and technical staff being represented in all leadership groups, right? Everybody, all of the stakeholders, everybody who's doing work in this organization, their voices need to be heard, and we have to make sure that they are represented in the decision making. So just a note about how we make decisions. We use the sociocratic method. Again, this is a way we chose that worked for us. I think some of the features that I really like about it are that the idea is that anyone can have an idea. So anybody might have an idea about how the organization might work better. Then a group of stakeholders, often it's a working group because it might be something scientific, like the physiology working group might want to have a proposal for how a certain set of data will be collected. They make a proposal, they actually work out a draft. We talk about that across the organization, either in Slack, sort of across Slack, across comments in Google Docs, in Zoom meetings, and then the policy basically is edited in response to this discussion. And when the working group thinks it's ready, it will go to the General Assembly for a vote. But I want to point out that this is not just any vote. We're asking folks to tell us whether the proposed course of action is good enough for now, and safe enough to try. I mean, you can imagine that when 20 PIs are voting on whether this is a way to proceed, the idea that all 20 will agree that this is what they would like is like 0. But if everybody can agree that this is, we trust the stakeholders, right? The working group who are really involved in making this decision and who are really involved in its outcome, that we can trust them and that they have an idea that is worth trying and is not going to tank the organization, then we can move forward. And I should say this is an iterative process. We learn through experience what works, and when it doesn't, we start revising. So this is like a constant loop. And in fact, we're actually on our 3rd version of the authorship and credit assignment policy. As everyone has talked about so far today, that is like probably one of the toughest nuts to crack in team science. Okay, so I want to talk just briefly about policies in general. With apologies to Robert Frost, I think that setting clear expectations about how we work together in advance, and this goes back to, I think, what Alyssa was talking about, sort of making sure that collaborations, as they come together, have agreements about how things are going to work, is super important. Right? It builds trust and confidence in the organization. It protects researchers against power imbalances. It creates a lab culture that improves working conditions for all. And I think that in general, labs can benefit from a lab manual to clarify norms and expectations. I think this is maybe not so, this general idea is maybe not so revolutionary, but when you are bringing together a bunch of labs that all have different lab cultures, ensuring that there is one agreed upon culture of your joint group, of your team, becomes really critical. And again, policies should be updated regularly with lessons learned. So some areas where we have formal policies, conflict resolution and ethical behavior. This is really critical for cross-institutional collaborations. You might not think about it that much, but in fact, all of our institutions have ethical codes, often have conflict resolution policies, but they may differ pretty dramatically. And you want to make sure that everybody is in agreement about how we work together and how we resolve conflicts. Authorship and credit assignment, obviously pretty tricky. Data release and sharing. And on our website, we actually make all of our policies publicly available. So if you have any interest, you can go to our website and check them out. Okay, so now I want to dig in a little bit on authorship and credit assignment, since I think this is something that is particularly, we have wrestled with a lot, and I think is relevant to many, many groups. So this is actually the first, our first big paper that we wrote was this development of a standardized and reproducible measure of decision-making in mice. And you can see that the author list is pretty long, maybe not as long as some of the ones that Liz showed, but plenty long. And the different work was done by each of them. And I'll show you a little bit about how we use the credit, the credit policy, taxonomy rather, to differentiate this. But I also want to draw attention here to one thing that what became part of our policy was the idea of a task force. So although many people contributed to the data collection, to the analysis, to data curation, all kinds of pieces of the taxonomy, there were a small set of people who actually wrote this paper, who wrote most of the text, who created the final figures, and who really like shepherded the work from the idea to a published paper. And those people we call the task force, and they are recognized, especially in the taxonomy and also in the acknowledgements. We try and really highlight that these people did an important piece of the work. So I think it's not so important sort of what this is, but we shared the method, we shared the data set. But you can imagine that with this much, with all of this, there were a lot of people involved. Okay, so this is what the taxonomy looked like for us. We published this paper in eLife, and they do actually use the credit taxonomy. So the text itself and the metadata went into the eLife site in the regular way. But we also wanted to give a graphical representation, because with so many people, it's hard to maybe make sense of how all the people contributed. And so we created this matrix. There are other versions of this. We did not invent this in any way. I don't want to say that, but we did create a sort of graded version so that you could see across the top are all the authors, along the side are all the taxonomy categories, and then the colors represent basically how much of a contribution. It's mostly sort of high, medium, and low, and people basically graded themselves in how much they had contributed. I like this because I think it gives a sense for how the work that everyone did came together in this group to produce the paper that we published. Okay, I will say, however, that when we looked at the people in the organization and the list and the sort of the classic credit taxonomy, which I love and which I think captures a lot of what goes into writing the paper itself, there were a lot of contributions because we're a large group, sort of making that large group team science work. There were a lot of contributions that didn't fall into this list. So, for example, outreach, sort of making sure that the work that we did was, and the data that we share and the tools that we share are accessible to the community and that they're aware of them. Funder relations, developing the pipelines that were necessary to get the data brought into the data architecture. Project management, to manage across all of those different labs. Technical support within the IBL, technical support outside the IBL for all of the things that we share. Creation of the data architecture, code review. We needed to make sure, since all the code was being written by different people, but we wanted to release the code. That's another piece of our open science. Someone had to look at it, right, and make sure that it was ready for release. The paper itself needed to be reviewed internally before it was ready to go. All the governance and policy work that goes on, all the scientific planning. We have an annual meeting, so there's conference planning. And finally, all the leadership that is necessary. As you saw, there's all these small groups and many people are leading those groups to make that work move forward. So there are many new kinds of contributions that are required for successful team science. And I just want to point out that we want to make sure, who is doing this work and how do we advance their careers. So the solution, I'll say very briefly, the solution for the International Brain Lab is that some of this work is done by postdocs and graduate students and PIs, but we also have full-time professional staff who do much of the technical support, who manage a lot of the data architecture. So all of the infrastructure, many of the things that basically are going on in the column on the right are being done by the staff, and they are a really, really key piece of the IBL's success. Okay, now one more piece on authorship and sort of that leads into the next sort of way that we have been thinking about team science. And that's that the authorship policy, one of the early adaptations of our authorship policy was actually about inclusion. So we had originally chosen an opt-in model, and we felt this was the fairest approach. If you could fill out an author contribution statement in good faith, you could be an author especially on one of these large papers. But we realized that this left too much room for individual judgment. We couldn't only rely on people to think their contributions were enough, as some, particularly those who have a history of being marginalized, have a tendency to underestimate their own contributions. And this was a red flag generally for us, that as much opportunity as our sociocratic governance offered for folks to express opinions, right? Remember, I said everybody can have ideas and we can turn those ideas into policies. We were still missing a piece that enabled us to ensure that all voices were being heard. So to address this, we do regular anonymous surveys, and we report out the results to the entire collaboration. So this is an example of what the survey might look like. It's pretty straightforward. We administer it annually to all members of the collaboration. We ask questions about all aspects of lab culture, about expectations, about relationships with the PIs, with peers, with technical staff. We ask specific questions about experiences or observations of harassment or bullying. And we report these results to the entire collaboration. And it's nice because it basically seeds new proposals for cultural change in the lab. It allows us to basically, to be iterative in our approaches to improving how our team science works. So I just, it's an open science thing. credit where credit is due, this was adapted from a great tool by Leslie Boshall, which I think can be useful for any lab, and you can find it online. So finally, I want to talk a little bit about communication. It goes without saying that the bigger the group, the harder it is to ensure good communication. This is particularly true for us as a distributed collaboration because the usual methods of knocking on doors or meeting up at the coffee machine don't always work. And we found that knowing where to find specific kinds of information or how to ask for it can go a long way. So luckily, there are a lot of tools out there to facilitate communications in organizations, particularly those that are distributed. I think post-pandemic, nothing I show you is going to be something you haven't seen before. And we've just taken advantage of basically each of their strengths to create a communications toolbox. But I really want to say that ultimately the choice of tools really depends on what people are most comfortable using. The best tool is the one that people will actually use. And there is like, even if the tool is like perfect for your setup, but nobody wants to learn it or it's too hard to learn, it won't work. It's not useful. So we happen to use G Suite for documents and for ratifying decisions. We use Zoom to discuss and delegate face-to-face. We use Slack for spontaneous and synchronous communication and real-time interactions. And finally, we use GitHub for technical sharing. So a last note about building and maintaining teams. You have so many people, you have lots of ideas and activities, and as in this like kind of kooky graphic, not everyone's going to be talking with everyone else. So it's really important to have mechanisms for building and maintaining organizational knowledge. So onboarding is really, really key here. In many labs, I think onboarding can be a little bit haphazard. People arrive in the lab and they kind of get taken under the wing of somebody and then they learn what they need to learn. But in a large and distributed organization, that can be a challenge. So you want to get people up to speed quickly. Whatever you design, you want to focus on getting people up to speed quickly. You want to clarify where to find resources and who to ask for help, right? These are sort of the key pieces. Relationship building, I think Alyssa also pointed to this, like the relationships among people is really what helps the team to work. And so finding ways to build those relationships, particularly in distributed teams, is really important. We do this through annual meetings, through hackathons, through lab meetings, which are virtual, and through trainee visits, where for example, we try and get experimentalists to go and visit theorists to like spend some devoted time to working on a project, or send theorists to an experimental lab to really see how the data is collected. Finally, we do team meetups wherever we can. So if people are at SFN or at Cosine or at the Simon's annual meeting, it's an opportunity for everybody to get together, to go out to dinner or to go for a drink, and just to sort of cement those relationships whenever we can. And finally, I'll say documenting, you have to document everything. That is really, really key. So we standardize meeting agendas and minutes as a way of trying to ensure that we have some set of information that comes out of every meeting. We record our Zoom meetings in case people want to go back and look at them. And I think one of the things that we have struggled with, but is super important, is making everything easily accessible through whatever mechanism that you choose for your team. So finally, I will say, again, to echo some of the things that have been said earlier, hopefully we as a team are better than the sum of our parts. And I think the things that have made our team science productive have been strong relationships and trust among teammates, representation for all team members, clear decision-making processes, policies that reflect organizational consensus, mechanisms to ensure all voices are heard, building, maintaining, and sharing organizational knowledge. And I want to say that these best practices facilitate teamwork, but I think they also improve the quality of the workplace for everyone. And so I really hope that at least a few things I said today resonate with you, and maybe you can take them with you. And finally, I want to say thank you. The IBL is enormous by many standards, but everybody has contributed so much. And of course, thank you to our funders who have very generously funded this experiment. And with that, I'll say thank you in case there are any questions.
Thank you so much, Hannah.